तुमच्या याच्यावरनं जो पीसीचा आवाज आहे ना आपलं लॅपटॉपचा तो वाढवा सर थोडासा.
येस. ओके. येतोय
नाही सर कमी येतोय पण कमी येतोय सर.
आता येतोय जास्त कारण वाढवलंय
सर. नाव वी आर लाईव्ह नाव YouTube.
ओके ओके.
येतोय आवाज आता?
कमी येतोय सर पण येतोय. प्रशांत तुला येतोय का? मला कमी येतोय आवाज येतोय?
हो सर येतोय बट कमीच येतोय म्हणजे क्लिअर नाहीये.
कालचं हे घ्या ना सर तुमचा कालचा जो हे
घेऊ?
होता ना
ओके.
हेडफोन कालचा.
डमाले सर.
अलाव एव्हरीबडी नाव. प्रशांत सर.
सर केलंय अलाव केलंय सर.
आलेत ना सर?
हो.
ओके पार्टिसिपंट सर फुल ओके. someone check for YouTube.
केलंय सर.
दमाले सर तुमचा आवाज नाही येते.
Hello.
हा येतोय सर.
Good morning all the participants. Welcome to day 2 of this national level online faculty development program. I am audible to all?
Yes sir, yes sir. Just 2 minutes. Sir.
Sir, just tell me when to start.
Okay, yes. Once. So, meanwhile, you are having your PPTs ready. Prashant sir.
Yes sir.
College. I'm asking regarding college. Okay, let it be. Sir is getting ready.
Hello. झालं.
All participants please turn off your videos.
Yes sir. Instructions.
Participants please turn off your videos.
Okay. Good morning all. Good morning all the participants. I request all the participants to kindly mute their audio and stop their video. In order to have better bandwidth, please turn off your video and mute your audio. Welcome to this day 2 of national level 1 week faculty development program. Should I start, sir?
1 minute. I'll just check it whether sir is able to access or not. Sir, it is audible now?
Yes.
Okay, sir. You need to just put it up your mic. Still the voice is little bit low, but nevertheless we will move forward. थोडंसं वरती लावलं तरी चालेल सर तो तुमच्या याचा. If you are comfortable with the mic, that is also okay, sir. हा.
येतोय आवाज आता?
आता येतोय सर. फक्त ते वरती लावलं.
Yes, sir. Audible. No problem.
Okay.
वरती लावता येईल सर कलर वगैरे कारण बराच वेळ बोलायला लागेल.
Hello.
Okay. Yes, yes, it is fine now. Good morning, one and all present for this EFDP on insight of basic techniques and recent advances in pharmaceutical sciences and research and application perspective. I welcome you, sir, on behalf of Srinath College of Pharmacy and Bharti Gramin Punarachna Sanstha in association with IPA. Welcome you, sir. Over to you, Damale, sir.
Yes, thank you, sir. Again, I welcome all the participant, resource person for this day 2 of national level online faculty development program entitled Insight of Basic Technique, Recent Advances in Pharmaceutical Education and Research Application Perspective. So in this day 2, we have two sessions. First session is regarding regulatory perspective and second session is regarding application of spectroscopic technique, FTIR and NMR. So let me begin with the first session, the
Slide,
day 1 session.
Prashant, sir.
So day 1 session, for this day 1 session, we have a resource person, Professor Dr. Nagesh Alulkar, sir. Sir is currently working as a professor and principal at Satara College of Pharmacy, Satara. Sir also sharing an additional responsibility as a Dean of Pharmacy at Dr. Babasaheb Ambedkar Technological University, Lonere. Sir has more than 21 years of teaching and research experience. Sir is recipient of facility extended by intellectual creation and diffusion services of National University of La Plata, Argentina. Sir have received more than 21 lakhs of research grant from AICT under FDP and RPS scheme. Sir has published his research work in more than 53 research articles, has one patent and one book authorship to his credit. Sir has presented his research work in more than 20 national and international conferences. Sir has acted as a resource person for more than 24 workshops, conferences and faculty development program. Sir has guided more than 48 MPharm student and one PhD student. With this brief introduction, I kindly request Professor Dr. Nagesh Alulkar sir to please take charge of this particular session and start the session. Over to you, sir. Please start the session. Prashant sir, please stop the sharing.
Yes, yes.
Stop your sharing. Okay. Now you can share the screen, sir.
Yes, just enable it because it is still disabled.
Prashant sir, it is disabled. तिथून multiple access द्या ना
Yes,
खालून.
yes, sir. केलंय, sir.
Now it is okay, sir.
No, still the message is there.
No, now it
Try
is
again, sir.
working.
Sir,
It
try
is
again.
working now, sir. Try again, sir. Yes, sir. Slide share, sir. Okay, sir. Fine. Now it is.
Okay.
Yeah, full screen is okay, sir.
Good morning to one and all. Thank you for the nice introduction, Damle sir. Today I am going to deliver a presentation on IND and ANDA regulatory perspectives. So everybody is well aware of the fact that how a new drug is developed and how much time and how much money is invested by a company for development of a new drug. So this is a flowchart that is given, and this particular figure represents how the different phases are involved in the development of a new drug. Most of the students or most of the faculty have learned it during their college days, but still, for this presentation, I have taken it in brief that how many different steps are involved in the development of new drug by any company. So this is something I have taken it for your references. So different phases I have taken it: preclinical search, clinical studies, NDA and review. Today's my most of the talk will be concerned mostly with ANDA, that is abbreviated new drug development. But before that, everybody should be well aware of the development of a new drug. Therefore, this particular slide is taken for your information. So phase 1, phase 2, phase 3, and you can see the short-term, long-term processes that are involved in the development of new drug. and how and when the FDA is coming into picture. So the different bars, the figures, the arrows are given in this figure representing when and where the FDA is coming into picture, when and where the company is coming into picture, and when and where the how much time is taken by the industry and how much time is taken by the FDA. So this blue line indicates the time taken by FDA. The white portion indicates the time required for the industry, and see the pink, particularly the pink triangles. So after the carrying out the synthesis and purification and animal testing, the IND is submitted to FDA. Then again, the blue line indicates that the FDA is taking time for review. And once the review is finished, then the permission is granted by FDA for carrying out the clinical studies. So once again, the company comes into picture that phase 1, phase 2, phase 3 clinical trials will be carried out, and the NDA is submitted to FDA. Once again, the FDA is taking review and giving the permission to launch the product. Once again, see here, the meetings are carried out with the sponsor and FDA. These diamonds, yellow diamonds indicate that whenever the meeting is being conducted between the sponsor and the FDA, these yellow line, yellow triangle, particular diamonds are showing that the meeting is conducted between a sponsor, that is a company, and the FDA. And these green triangles are indicating that the advisory committee, the meetings of advisory committee are being conducted, and the reports are given to the given by FDA to the company. And based on all the what we can say, the formalities being completed by the company, and if FDA is satisfied, then and only then the new drug is brought into market. But everybody is well aware that to bring a new drug into market, it requires several hundreds of new chemical entities to be manufactured to be synthesized in the laboratory. And out of several hundreds of new chemical entities being synthesized in the laboratory, only one may hit or may not hit the market as a new drug. So there is a difference between a new chemical entity and a new drug. There are hundreds of new chemical entities that are being synthesized in the laboratory, but only one, or may not be one also, may hit the market as a new drug. So something that is associated with the new drug development, the few terminologies I'm taking here for your reference, that is treatment IND. A treatment IND or a treatment protocol permits the use of an investigational drug in the treatment of patients not enrolled in the clinical study. So this treatment IND is filed by a company. So this treatment IND or a treatment protocol permits the use of an investigational drug in the treatment of patients not enrolled in the clinical study, but who have a serious or immediate life-threatening disease for which there is no satisfactory alternative therapy. You can take the case of COVID-19 right now, because there is no as such treatment. Every company is trying to bring the medicine for COVID, but so many companies are trying for vaccines too, but still, though no therapy has been completely foolproof therapy has been brought by any of the company in the interest of the public or in the interest of the society at large. Therefore, treatment IND. IND for an orphan drug. Under the Orphan Drug Act, an orphan disease is defined as a rare disease or a condition that affects fewer than 2 lakh people, and for which there is no reasonable acceptance that the cost of research and development for the indication can be recovered by sale of the product. Therefore, for orphan drugs, the special provisions are made by the FDA, and special provisions are taken into consideration. So what that special provision is, incentive to orphan product development is a provision of a 7-year period of exclusive marketing rights after the regulatory approval of a patient. So this 7 years time is given by FDA particularly to incur the expenses that are being invested by any investing the company for bringing up the orphan drug, because the total number of population that is suffering from a particular disease is less than 2 lakh. So whatever the cost that is incurred by the company has to be recovered by the company. Therefore, this special provision is made by the FDA, and the incentive is provided of 7 years. Then after IND, the part is new drug application. So these are steps. First is IND, second is NDA, and finally is on ANDA. So most of the talk will be dedicated to ANDA. But before ANDA, I will be explaining you what IND is, what NDA is, and then I will be coming towards ANDA. Third part. So what is new drug application? If the 3 phases in clinical trials or clinical testing during the development period demonstrate that sufficient drug safety and therapeutic effectiveness has been achieved the sponsor may file for an ANDA with the FDA. So what the new drug application is and what FDA is carrying out. So FDA is taking out most of the measures for approving the new drug. So what a new drug classification is. So new drug for which we can call as a new drug. So NDA, that is called a new drug application or NDA. The classification is as follows. When a new molecular entity is proving that it is safe and efficacious, then you can file a new NDA that with the FDA. If a new salt of a previously approved drug, but not a new molecular entity, you can call it as a new drug also. A new formulation of previously approved drug, not a new salt or a new molecular entity, to which you can also call as a new drug. You can file a new drug application. New combination of 2 or more drugs, you can file a new drug application with the FDA department. A new indication or a claim for already marketed approved drug, so that is including switching over from marketing status from the marketing status of the drug from prescription to OTC can be covered under NDA. An already marketed drug product, if it is there in the market but no previously approved NDA is there, you can file the NDA to it, the FDA. And what are the different fundamentals that are associated with NDA submissions? So this is the different, what we can say, the different things that are required by to be submitted by a manufacturing company or an investigator to the FDA. It contains index, summary of your inventions, then chemistry, manufacturing and control, samples, method validation, packaging and labeling, non-clinical pharmacology and toxicology. Fundamentals of NDA submission also includes human pharmacokinetics and bioavailability studies. Microbiology, it is specifically required for antimicrobial drugs only. Then the clinical data and safety update report that is typically submitted 12 months after the NDA submission. It also requires to be submitted to the investigator company the statistical data that is generated after the during the development of new drug. Then case report tabulations, case report forms, patent information and patent certification. All these things are needed to be submitted by the investigator company to the FDA. So this flowchart explains how the process of NDA is moving ahead. So NDA review process, so applicant is filing, it is called as a drug sponsor, it is filing a NDA and to the FDA department. After filing the NDA to the FDA department, so the FDA decides whether the application is fileable or non-fileable. Whether I'm audible? Hello?
Yes, sir. Yes, sir. Yes, sir.
Yes, sir. Yes, sir. You
Whether
are
I'm audible?
Yes, sir. Yes.
Hello?
Yes, sir.
Sir, you are audible, sir. You are audible, sir.
Okay. So application, whether it is fileable or non-fileable. If the application is fileable, it moves ahead. If the application is not fileable, then the FDA refuses to file a letter. FDA sends a letter for refuse letter to file NDA to the drug sponsor company. If it is fileable, then the review will be taken by Center for Drug Evaluation and Research, and which different reviews are being carried out. The reviews based on medical, biopharmaceutical, pharmacology, statistical, chemistry, microbiology, all these reviews are carried out by FDA. And if suppose during this review by CDER, if the review qualifies, then the FDA asks the sponsor company to file the reviews complete. And if it is complete and acceptable, then the labeling review will be taken place and inspection of site. So whether labeling reviews and both inspection of a site is conducted successfully, then the NDA action will come in, or NDA will be granted by the FDA. If suppose during this process, FDA is not happy with whatever data you have submitted, then in that case, FDA will conduct the advisory committee meeting. Then meeting with the sponsor are carried out, and suppose some reviews are to be taken. In that case, the FDA asks the drug sponsor company to carry out the submit the application once again. Sponsor revises it, and additional information if it is required, the FDA asks the drug sponsor company to submit the additional information. And after FDA is satisfied that whatever the information that the company has submitted, the drug sponsor has submitted, and after the complete satisfaction by the FDA will grant the new drug application to the company. So now towards, if suppose there are again alternative supplemental new drug application, a sponsor of an approved drug NDA may make changes in that application through the filing of a supplementary NDA. Depending on the changes, some require FDA approval before implementation, whereas others do not. The changes that require prior to approval for supplemental NDA, that is use of a different facility to manufacture the drug substance where the facility has not been approved through inspection for CGMPs within the previous 2 years. It is a case for filing the supplemental NDA. If suppose you are shifting the facility of manufacturing from one place to other place, and that place is not inspected by FDA, you have to submit a supplemental NDA. So use of a different facility to manufacture the drug substance where the facility has not been approved through inspection for CGMPs within previous in the last 2 years or previous 2 years. In that case, supplemental NDA you have to file. A change in the method of synthesis of the drug substance. If suppose you are changing the method of synthesis of the drug substance, in that case too you have to file supplemental NDA. Third condition where you have to file supplement NDA, that is a change in the formulation analytical standards, method of manufacture, or in-process controls of the drug product. If this is the case, again you have to file a supplement NDA. Then use of a different facility or a contractor to manufacture a process or package of the drug product. In that case too, you have to apply for supplement NDA. Change in the container and closure system for a drug product. If it is the case, supplement NDA comes into picture. Extension of the expiration date for a drug product based on a new established data. In this case too, you have to file supplement NDA. Then any labeling change that does not add to or strengthen the previously approved label statement. In this case, you have to file, the company has to file supplement NDA. Now come towards the important part of today's presentation, that is ANDA, that is called as abbreviated new drug application. Most of the people are well aware of the fact of generic drug because we have come across the fact of generic drugs through Amir Khan's Satyam Jyoti program or everywhere because it is not a new concept to the pharmacy people. Generic drug, everybody is now discussing about generic drugs. Our government has also started some generic medicine shops under Bharatiya Jan Aushadhi Pariyojana scheme. Then what is generic drugs? Most of these people who are belonging to pharmaceutics or politicians may be knowing about generic drugs, but the people who are not associated with pharmaceutics particularly may be knowing something or may not be knowing something. So today my presentation will be giving some insights into this particularly abbreviated new drug application or the generic drug. A generic drug product is one that is comparable to an innovator drug product. It is also known as the reference listed drug, RLD, as identified in the FDA's, that is US Food and Drug Administration's list of approved drug product with therapeutic equivalence evaluations. As far as dosage form, strength, route of administration, quality, performance characteristics, and intended use are concerned. So we have to copy the reference listed drug and you have to produce the results comparable to the reference listed drug that is present or that is present in the orange, given in the orange book. So ANDA is the abbreviation for abbreviated new drug application. It contains data which when submitted to FDA's CDER, that is Center for Drug Evaluation and Research Office of Generic Drugs, provides for the review and ultimate approval of a generic drug product. Once approved, an applicant may manufacture and market the generic drug product provided all issues related to the patent protection and exclusivity associated with RLD has been resolved. Because whenever we are filing ANDA, we have to take into consideration the patent filed by the brand holder company, because every company, when it is bringing any new molecule or new drug into the market, it is patenting. So patenting is particularly we are taking some rights with us, and we are doing it because nobody should market whatever the investment we have done. We are taking the patents and we are having the patent for protecting our intellectual property and gaining the money from whatever we have invested in development of a new drug, because thousands of crores of rupees and more than 12 years of time is required for bringing a new drug into market. And if suppose if it is not patented, then anybody can take the benefit of the situation. Therefore, this patent provision is there. And whenever we are filing the ANDA, we have to take into consideration that the patent is nowhere infringed. If suppose we are not taking into consideration the patent infringement, then we have to suffer a lot. Therefore, we have to take into consideration whatever the patent protection rights are issues are concerned, and the information provided in the generic about the branded drugs in the Orange Book. The generic drug applications are termed abbreviated. The basic difference is between the new drug and the abbreviated new drug application is that we don't have to carry out so many things for abbreviated new drug application, or particularly for the application of generic drugs. Because why they are abbreviated? The term abbreviated, because abbreviated in that they are generally not required to include preclinical and clinical data to establish safety and effectiveness. Whenever a new drug is brought into market, the company has to invest tremendously on clinical trials. But here, as the safety and efficacy both have been established by the sponsor company or the brand holder company, because the brand holder company has carried out both the preclinical as well as the clinical data for establishing the safety and efficacy of the new drug, the abbreviated new drug application is not required to carry out that study once again. Only the bioequivalence studies to be carried out or to be conducted, and based on the bioequivalence studies, the abbreviated new drug application can be sanctioned. Because these parameters were established upon approval of a innovator drug product, which the first version of the drug product is approved by the FDA. So when this ANDA is brought into picture, so we have to take into we have to have the peep into the history. In the beginning, there were no legal generics. The 1906 Act of the drug law, it was just giving highlight, it is giving emphasis on just misbranding and adulteration of the drugs. And no new drug application provision was there in the Act. In 1938, the Act introduced the new drug application provision. At the time of 1938, it did not directly address the generics. What was the marketplace reality at that time? Not new drug rulings up to early 1940s. Several Me Too's entered the market. It was the era of Second World War, and there was need of the medicine. So several Me Too's we can call as IRS, that is identical and related, and that is similar drugs that entered into the market. We call it as IRS drugs, identical, related, and that is similar drugs. Several Me Too's, they're particularly termed as Me Too's, that entered the market. And basically it was the era of Second World War. The Second World War between that was happened between that is in the years from 1939 to 1945. And during that time, the entire world was in need of the drugs, and that was taken up as a chance from the companies, and they brought into market several Me Too drugs. But what happened that in 1962, the act was passed. It is called as Kefauver-Harris Amendments. This particular Kefauver-Harris Amendments came into picture, taking into consideration the tragedy of thalidomide. The thalidomide that was marketed into that was the marketed drug, but unfortunately, the thalidomide has created a havoc in the market. And after the thalidomide incidents, the U.S. Congress has set a committee under the Kefauver-Harris, and they proposed the amendments in the act, and these amendments are very famous as Kefauver-Harris Amendments, and these amendments were included in the year 1962. Till then, only safety of the drug was taken into consideration. They have added efficacy, and drug efficacy study implementation was introduced. This is also called as DESI, and so many drugs that were marketed from 1938 to 1962 were asked to conduct the efficacy studies. And this particularly Kefauver-Harris Amendment has brought us a new change into the scenario into the global market. And along with the safety, the efficacy issue was also added by Kefauver-Harris Amendments. So it has laid down the foundation for the first approved generics. By 1970, the ANDA was created. But the problem was that only applied to pre-62 drug that were found effective under DESI, that is drug efficacy and state safety indications. Then when the modern generic industry took the birth, it was year 1984. From 1962 to 1984, several things have taken place. And in the year 1984, before 1984, US Congress has set one committee under the guidance of Hatch and Waxman, Senator Hatch and Senator Waxman, and they have carried out the investigation in the entire system and put forth the amendments once again, and this is very famous as HatchWaxman Act. This act is particularly has given a boost to both the innovators as well as the companies which are interested to file the generics with the market. This particular act is called as Drug Price Competition and Patent Term Restoration Act of 1984. It was brought, or it was enacted on September 24, 1984. This Drug Price Competition and Patent Term Restoration Act, this particular act was brought into 1984. What were the objectives of this HatchWaxman Act? Whenever we are talking about ANDA, this HatchWaxman Act is to be studied in detail because this has laid down the foundation of modern generic drugs, modern generic industry. So what were the objectives of HatchWaxman Act? So first is to reducing the cost associated with the approval of a generic drug, because before that, whatever the cost that was associated with the development of a generic drug was very high. And HatchWaxman has taken into consideration that the drug particularly should be affordable, particularly generic drugs must be available to the population at large, and the cost of the medicine should be affordable to the common public or society at large. So their first objective is to reduce the cost associated with the approval of a generic drug. Then second was allowing early experimental use of the patented drug. Then compensating the branded drugs manufacturer for the time lost from the patent term because of the regulatory approval formality. We know that it takes a lot of time for patent to be granted, and take into consideration the time lost for a patent brand company. The HatchWaxman Act has provided some kind of exclusivity as a promotional or what we can incentive to the brand company. So it is a compensation provided by HatchWaxman Act to the innovator company. Then motivating the generic drug manufacturers to bring into the market the cost-effective and beneficial drugs. So HatchWaxman has Act particularly balanced both the innovation as well as affordability. How innovation? By providing incentives to the pharmaceutical companies to research and develop new innovative pharmaceuticals, because new drugs, whenever a new drug is coming to market, new diseases are also created, new diseases also come into picture, and for those new diseases we also require new drugs. And if suppose no incentive is provided and no patent is there, then automatically no company will be there, and taking into consideration, no company will be there to investigate or invest money on innovation. So taking into consideration that particular aspect, HatchWaxman has provided some incentives for innovation of the for the manufacturing companies who are investing huge money for innovation. At the same time, it has taken into consideration the large population, because affordability, because the most of the people should be in need of cheaper and effective and safe medicines, and taking into consideration the affordability was taken into consideration. So affordability by creating an abbreviated path for generic manufacturers to bring lower cost versions of the pharmaceuticals to the market. So this is the balance between innovation and affordability. So what are the general provisions of HatchWaxman Act? So HatchWaxman Act added 2 provisions to the Federal Food, Drug and Cosmetic Act, namely Section 505B2 and 505J. Section 505B2 was added with respect to hybrid new drug applications, and section 505J, which was added with respect to abbreviated new drug application. So 505J we are discussing. So HatchWaxman basics, what are the requirements? We have seen in the introduction to the ANDA, what are the requirements of filing for the ANDA or generic drugs. So the basic requirements, it is the same active ingredient, same conditions of use, that are labeling conditions, same dosage form, same strength, same route of administration, bioequivalent, patent certification. So all this should be compared with the reference listed drug, that is a brand new product. That is. Then what is abbreviated new drug application? ANDA is the abbreviation for abbreviated new drug application. It contains data which, when submitted to Center for Drug Evaluation and Research, Office of Generic Drugs, provides for the review and ultimate approval of a generic drug product. So once approved, an applicant may manufacture and market the generic drug product, provided all issues related to patent production, safety, and efficacy has been proved by the generic drug manufacturing company. Generic drug applications are termed abbreviated because they are generally not required to include, just we have discussed earlier, that preclinical and clinical studies data to establish safety and efficacy, because it is already established by the brand new company, brand holder company. So a generic product, we know that it is one which is comparable to an innovator drug product in dosage form, strength, route of administration, quality, performance, characteristics, and that is the use. The generic drug applicants must scientifically demonstrate that the product is bioequivalent. What is bioequivalent? It should perform in the same manner as that of the innovator drug. So the basic requirement is establishment of bioequivalence, that is bioequivalence data, because rest of all the things we are giving, but we are not carrying out the clinical and preclinical studies. The emphasis is given on bioequivalent studies by HatchWaxman Act, and any generic drug manufacturing company has to establish the generic drug the bioequivalency study and prove that your product is bioequivalent with the innovator drug or a reference listed drug product. So the rate of absorption and or bioavailability of the generic drug is equal to the innovator drug. So what is the key point? So in order to submit a NDA, there must be a RLD, that is a reference listed drug. Where you can find the reference listed drug? So reference listed drugs are approved drug products with therapeutic equivalence evaluations, and they are present or they are given in orange book, and they are denoted with a plus sign. So you can have this electronic orange book. The website is provided here, and this orange book you can search and you can find out the reference listed drug. So many things are associated. I will not discuss it here. Electronic version of the orange book is also there. So just what is the difference between the brand new drug and generic drug? So only you can see first 5, all the things are same. From 6, 7, 8, the change is there. So for NDA, it is animal studies, clinical studies, and bioavailability is required. And whereas in case of generic drugs, only bioequivalence studies has to be carried out and established with the brand drug. So what is an option if suppose you are having some kind of problem in filing a generic drug or NDA application? There is a suitability petition, maybe an option available with the generic drug company. So suitability petition is a second option that is provided by FDA to the generic drug manufacturing company. You can file a suitability petition in a few of the cases. If suppose the NDA product is not identical to the listed drug in the form of either in terms of route of administration, in terms of dosage form, in terms of strength. If suppose one of the active ingredient in a combination of 2 or more drugs is substituted with another active ingredient, in that case, you can file a suitability petition with the FDA department. Instead of filing ANDA, you can file a suitability petition if suppose either of these 4 reasons you are carrying out some kind of changes, you can file a suitability petition. In the year 2003, PREA come into picture. What is PREA? That is that is a particularly Pediatric Research Equity Act. Whenever you are carrying out the clinical trials or based on in the children, infants, then they cannot give their consent. You have to take the consent of their parents. So this particular act came into picture, and while you are filing a suitability petition, this PREA is to be taken into consideration. According to HatchWaxman Act, the different certifications are there. What are those certifications? There are 4 conditions. These 4 conditions are called as certifications according to HatchWaxman Act. These 4 possibilities. First possibility, that the patent information on a drug has been lost. That is, no patent information appears in the Orange Book. In that case, you can file ANDA. If suppose the patent is already expired, no patent is existing in force, patent is expired, in that case too, you can file ANDA. Third, the date on which the patent will expire, you can file for ANDA earlier, and you can request to the FDA that the date on which this patent will expire, on the same day you can ask for generic drug approval. So this is a third case, third possibility for filing ANDA. And the most important, the ticklish and very interesting is the fourth parameter, the fourth possibility, that the patent is invalid or will not be infringed by the manufacturer, use or sale of the drug for which the ANDA is submitted. This is something which is challenging, but most of the companies are interested to file for para 4 filing. This is called as para 1, para 2, para 3 and para 4 filing. So whenever first 3 there is no challenge as such, but filing a para 4 filing, 505J para 4 filing, it is more challenging and more interesting too, because so many litigations are there, so many issues are concerned. A brand company comes into picture, filing a case against the generic drug company in the court. So many things are associated, and there is a challenge, and therefore most of the companies are. But if suppose you are winning the battle, you are enjoying a lot of benefits, therefore most of the companies are interested to file para 4 filing. These certifications are termed as paragraph 1, 2, 3 and 4 certifications respectively. So first, second, third, no such challenge as such, but fourth is really challenging. So paragraph 1, no patent is filed by any company, so it comes under 21 CFR 314.94. So many that is the sections. No patents referring the listed drug in the Orange Book. This is a condition for filing a paragraph 1 certification for ANDA. Firm is aware of non-listed patent, allows for immediate effective date of approval, and does not mean the patent cannot be enforced. So these are the conditions for filing ANDA under paragraph 1 or para 1 filing under 505J. If no patent is there into existence or in force, you can file ANDA under para 1 filing. Second type of filing, if suppose a patent is there but patent is expired, you can file for second. You can file the application for generic drugs under para 2 filing. The patent listed in the Orange Book is there, but though the patent is listed, the patent is expired. So you have to cite the patent number and provide the expiration date, and you can request the FDA for early approval of the drug. Then para 3 filing, patent is in force, but it is going to expire soon. In that case, you can go for para 3 filing. So the listed patent is in force at time of submission of your NDA. You have to cite the patent number and provide the expiry date. And you have to state the intentions not to market until the patent in force is going to expire. Once the patent is expired, then and only then you are willing to market or manufacture and market your generic drugs. This is called as para 3 filing. And this para 3 filing allows for approval immediately after expiration of the listed patent. So first 1 through 1, 2, 3, no challenge you can see as such for filing ANDA. Now come towards the challenging part, para 4 filing. So para 4 filing is challenging because we are challenging the patent of the innovator company. We are proving that your patent is invalid. The generic drug manufacturing company is saying that by filing NDA or generic NDA, we are no way trying to infringe your patent, or you are convincing that your patent is unenforceable. In these situations, real challenge is there for filing under para 4. Why? Because the patent is in force for so many days for so many years still ahead. So you are, when the patent is going to be in force for coming few years, still you are intent to market your product into market as a generic drug. So you intend to market prior to expiration of a patent of the reference listed drug or the innovator drug. That is the brand drug manufacturing company. Then you have to provide notifications, notification provisions to apply. A 45-day clock suit is provided to the brand holder company. Then allow for immediate effective date of approval if in case no suit is brought within 45 days. So I will explain which are the different steps that are involved under para 4. But if suit is brought within 45 days, because when a company is filing a generic drug application under para 4, it is sending the notice to the brand holder company that we are going to apply for generic drugs within 20 days. And a 45 days time period is given to the brand holder company to file a suit against the generic drug manufacturing company. If suppose within 45 days, the suit is brought by the company, in that case, FDA comes into picture and FDA locks your application for 30 months, that is 2.5 years. No ANDA can be approved by FDA for 30 months. If suppose a suit is not brought within 45 days of application, then automatically 180-day exclusivity is granted to the first ANDA filing company. But again, there is FDA is coming into picture. If suppose you are unable to file 505J, there is again 505B2 NDA for another potential option remaining with the companies. So, but still, though it is giving some kind of provision for generic drugs, it is again giving some kinds of statutory exclusivities under this act. So, which different exclusivities are given under that is HatchWaxman Act to the companies. Exclusivity for 5 years is granted under HatchWaxman Act for investigating the new chemical entity, and 4 years if NDA in para 4 challenge is there. 3-year exclusivity is given for NDAs, and FDA may not approve an NDA or 505B2 application NDA for 3 years after approval in case of new drug application. This also applies for new indications for switching over from prescription drugs to OTC. This kind of exclusivity is provided for 3 years. In case of orphan drugs, exclusivity is provided for 7 years. For pediatric drugs, exclusivity is provided for 6 months. And if suppose a company successfully, what we can say, facing the suit and giving the answers and easy to what we can say, that successfully completing all the pathways or all the procedures, 180-day exclusivity is granted to the first NDA filing company, provided all the matters associated with application are fully solved by the applicant. So patent and exclusivities we have just discussed. So what are the ANDA stability and batch requirements? So 3 months accelerated stability data must be submitted with the application. Then available room temperature set stability data should also be included. An update on subsequent room temperature data will be requested during the review process by FDA with the generic drugs manufacturing company. And one demonstration batch must be manufactured and submitted. You have to submit the source of bioequivalence study protocol. You have to submit the source of stability data, and the company has to submit the complete batch record for this batch to the FDA. So what is bioequivalence? The pharmaceutical equivalence whose rate and extent of absorption are not statistically, I am not saying, or it is not said that it should mimic. It should not be statistically different when administered to patients or subject at the same molar dose under similar experimental conditions. So AUC and Cmax at 90% confidence interval must be between 80 to 125% for proving the bioequivalence. Now, how to file a NDA with the FDA department? So these 10 points will explain how the ANDA is to be submitted to the FDA department. The signed FDA form 356H, the company has to submit. This provides information regarding the applicant name. As a DUNS number is provided, that is data universal numbering system is there. And this data universal numbering system, this particular number is to be provided by the company. Then name of the drug product, product strength, route of administration, indication of use, then proposed indication, a statement regarding whether the product is for prescription or over-the-counter drug. So this first part explains about this particular general information about your drug product. Then index should specify volume and page number for the complete and detailed items. After index, the information on the basis for which the ANDA is being submitted. This is important. Name of the reference drug, its dosage form and strength, information of the listed drug. If a suitability petition is approved, a reference to the FDA number that was assigned to that suitability petition. Then condition for using, including a statement regarding the condition for which the drug will be used, reference to the annotated labeling for the product, then statement of active ingredient, route of administration, dosage form, strength, all these things, because you are going to use the same things as that of the reference listed drug. Just you have to prove the bioequivalency. The labeling does not include just the label we are putting on the container or on the foil, but the inserts that are being prepared by the company is also the part of labeling. So including a copy of currently approved labeling for the listed drug as well as the proposed labeling for the drug being provided for the ANDA. Then chemistry, manufacturing controls, then human pharmacokinetics and bioavailability. This also include the information concerning to the design, dosing procedure, the number and frequency of blood and urine collection, and methodology for the assay. Then the samples, you have to submit the samples, 4 samples to the FDA, 4 different samples, and the FDA will carry out the tests, and question-based reviews nowadays available, and you have to go for submission of analytical methods for drug substances and drug products. Then labeling, 12 specimen of the final printed label, and all labeling should be provided by the drug product to the FDA. Then case report forms and tabulations. Then NDA review, as like NDA, again this flow chart explains the ANDA review process. The applicant is filing an ANDA with the FDA department. Then FDA once again goes to the application, acceptable and complete. If yes, then it is sending ahead for review by Office of Generic Drugs, Center for Drug Evaluation and Research. So this particular department is taking into consideration. Again, there are different groups of this OGD CDER. These different groups are taking to looking into this matter. Then they are looking for bioequivalence review, chemistry, micro, request for plant inspection, labeling review. If suppose all these things are acceptable and comply with the requirements of the FDA, the approval of inspection is acceptable, inspection is carried out, and if suppose inspection is perfect and complying with the conditions laid down by the FDA, ANDA is approved. In any of the situation, if nothing is proper, nothing is compliable, then FDA issues the letter for whatever the deficiencies that is found and asks for the company to comply with the deficiencies. At the first step only, if suppose the ANDA is not acceptable and complete, FDA rejects this and send the letter of refuse or issue the letter of refuse to the company. So applicant. So para 4 patent challenge, these are the basic steps that are involved. Generic para 4 filing. So generic drug provide notice to brand holder company within 20 days of acceptance. And brand must bring lawsuit within 45 days. And it is general procedure that the company which is having the brand, which is holding the brand, is going to court and it is bringing a lawsuit within 45 days. And automatically a lock period of 30 months is granted by the court, and that is no ANDA is sanctioned by the FDA, because generic company, if sued, automatic 30 month stay is granted to the brand and no ANDA can be filed. But suppose there is again a second window kept open, you can challenge it in the court, and again you can go for hearing. So generic manufacturer, it was brand name manufacturer, innovator, explaining existing innovating patent is either invalid or will not be violated by the generic drug manufacturing company. In reply, innovator can file a patent infringement lawsuit even if authentic claim by the generic manufacturer. If the suit is filed within 45 days, FDA doesn't give approval to the generic for a period of 30 months, that is 2.5 years. If suppose no suit is filed within 45 days, no action is taken by FDA. And after 2.5 years, US FDA decide if it should be granted approval or not. So these are some things which are associated with para 4 filing, because para 4 filing is challenging. Let us took a brief take into consideration the brief look into this, whatever that is the perspective as far as the generic drug application manufacturing is manufacturing and files are concerned worldwide, and what is the India's contribution in that. So over 150 products, including 20 blockbusters, having a sale of more than $77 billion in of total branded drug sales, and it's coming off patent in the coming 5 years. And there is a huge scope. And which countries had led most of the NDA approvals? First is Germany, second India, third USA, China, and Israel. You can see total 25% of the ANDA has been filed, or a particular approvals has been gained by Germany, 25% by India, 17% USA, 17% China, and 5% that is 17% by Israel. So finally, NDA approvals that are received by the different companies, different countries. USA 169. This is another some information regarding the ANDA approvals. So India 29% stake is there. Then ANDA approvals from 1999 to 2009, you can see the data, total number of ANDA filed and total number of approvals that have been granted by FDA. So day by day, if you see from 1999 to 2009, year-wise, the total number of applications for filing ANDA are increasing, and the total number of approvals are also increasing. Then which are the companies that are leading in as far as filing the ANDAs and gaining the approvals from FDA are concerned? You can see Glenmark, Aurobindo Pharma, Sun Pharma, Lupin, Dr. Reddy's. These are the front runner companies for grabbing the opportunity for getting the first ANDA to be approved by the FDA. So Indian companies backed 33% of, or 139 of 419 original ANDA approvals from US FDA in the year 2010. So patent challenge, that is para 4 filing by the countries. USA, the percent is 37%, India 21%, Israel 17%, and the rest of the countries are having near about 25 to 26%. stake. So if you say, what is the future perspective for the Indian companies? So taking into consideration, if you can see that the challenges as far as para 4 filing is concerned, and the 180-day exclusivity period is concerned, wherever and whenever it is found that so many problems are there, everybody is getting charged and trying to become self-reliant and trying to become more and more aggressive. It was thought that with the implementation of product patent regime, because in the year 2005 we have gone for product patent regime, and it was thought that most of the Indian companies will be engulfed by taken over by the foreign multinationals. But today what the situation is, Indian companies are there, rather they are being taken up by the foreign companies. These companies are going abroad or going out of the country, and they are taking over the foreign companies. So we can expect that in the coming years, we see that so many companies are, so many colleges are coming, so many mushroom. This is the particular talk of every academician, that so many colleges are being opened. But I'm always of the opinion that in the any case there lies opportunity, because it was thought that whenever the computer age came into picture, computerization, today you and me, we are talking only through this platform. This is the, what we can say, the IT revolution. Similarly, right from 1947, after the 70 or 74 years of independence, if you can see, the three particularly companies have brought laurels to our India. In 1947, we have to rely for life-saving drugs on most of the developed countries. Today, we are exporting, we are not only self-sufficient, rather we are exporting the drug to so many companies, so many countries in the world, and most of our revenue is coming from the out of the India, particularly from US. So don't be scared what will happen, no, whatever that happens, that always will happen in our favor, because India is a country of youths. Whenever any country has refused us to give supercomputer, India has developed it. Whenever any country has refused to give cryogenic engine, India has developed it. Whenever, suppose a condition will come, any country will charge for more amount, I'm sure that the scientist of our country is ready to develop the required drug for the newly generated or newly created, what we can say, any attack, either by the virus or by the bacteria or by any kind of disease. So don't be so panic. I'm always of the opinion that the youth of our country is self-sufficient to bring about the required changes. So we should be the first person to carry out the change in ourselves, and then expect our students to bring those changes, which will surely bring our country in the forefront as far as the pharmaceutical development is concerned, because only three fields right from the independence, first is IT, BT, and the pharma sector, ISRO and space research. All these things have brought laurels to our country. Pharma is one of it. I'm saying IT, I'm saying BT, or I'm saying ISRO, space research. Pharmaceutical innovation is not behind. Only what is required, as we have to be vocal for our local products, one thing. We have to become Atmanirbhar, but at the same time, after COVID-19, if it is expected that every company, most of the countries as well as the companies may be going from a global to local status. At the same time, let us expect that the Indian companies will be able to suffice the need of the Indian population at large, and will be able to suffice the need of the entire globe at the same time, and will be the front runner in the development in the coming years. So the take-home message for today's session is be vocal for the local products, be Atmanirbhar, and just expect that the Indian pharmaceutical companies will be the front leaders, front runners in the years to come. Thank you. Thank you for giving me the chance to interact with you. So thank you, Sharkey sir, for giving me the chance. And thank you, IPA Aurangabad branch, for giving me the chance to interact with the people on this IT platform or Zoom platform. Thank you once again.
Thank you, sir. Thank you. It was
Thank you,
wonderful
thank you.
session, sir. And per the box is concerned, there are so many questions. It means that it was an interacting and interesting session. Over to you, Karishma, for the question answer session.
Yes, sir. Good afternoon, sir. It was a very wonderful session. I'll ask the first question. It is from Deshpande, sir. What is the time frame for approval of ANDA application?
Please repeat.
What is the time frame for approval of ANDA application?
Yes, just I have explained that you have to file an ANDA with the particularly FDA department, and you have to give the notice to the brand company within 20 days. If suppose the NDA, the brand company is filing a suit against you for 30 months, no ANDA will be sanctioned. After that, and it is a general trend that the suit is being brought by the innovator company, and you will be blocked for 30 months. And after submission of ANDA, if suppose the FDA is taking the time, and once the FDA is satisfied, whatever the submissions you are done with the FDA department of the question-based review is there, the FDA is taking its own time. There are 3 different groups which are carrying out the activity of this particular application. And once everything is being satisfied by the generic manufacturing company, no patent is infringed anyway, FDA is taking its own time to sanction the patent, sanction the ANDA or generic drug approval. But suppose again, there are some loopholes of this HatchWaxman Act also. Like reverse payment system is there, branded generics are there, the companies are switching from OTC to prescription to OTC and trying to extend the patent. So there are so many loopholes, but still FDA will take its own time.
Yes.
Okay, sir. So the next question is, how long new drug can be considered as new drug? From Deshpande, sir.
Till the patent is in force.
20 years.
Okay, sir. Next question is, what is the role of bio waiver in ANDA, and what are the various bases for bio waiver?
So
This
bio
question
waivers-
is
Bio
Yes.
waivers are taken into consideration because whenever we are taking into consideration the liquid dosage forms particularly, not the solid dosage one. In that case, bio waiver, we can go for application bio waivers.
okay. Next question is, how the price of new drugs are considered? This question is from Shubhangi Iyer. How the
You
price
were-
of new drugs?
No,
Hello?
am I unable to hear?
Your voice was a little disturbing, Karishma madam.
How the price of new drug are decided?
Please ask the question once again.
So he's saying that how the price of new drugs are decided.
Hello.
Hello.
Yes.
So she is saying that how the prices of the new drugs are decided once it comes into the force. Once
How?
it is approved. How the prices of new drug,
Prices of new drugs.
those are decided.
No, for different countries, the different things are there. For India, DPCO is there, but for different countries, the different concepts are there. So whatever the money that is invested and how much, what we can say, the profit you want to earn, depending on that, the price is there.
So she
In
basically
in our country, the DPCO is there, drug price control order.
yes. They just want what is the role of DPCO inside that. They want to know that
No,
what is the role of
DPCO,
DPCO.
DPCO is a government agency which fixes the prices of the drugs. Because
Excellent drugs,
in our country, the government is taking into consideration, particularly because taking into consideration the population of our country, and the prices of the medicine should be within the reach of the pocket of the common man. The DPCO is there.
Yes. Any questions, madam?
Yes, sir. The next question is, on which criteria does the FDA classify NDA? By Poonam Raut ma'am.
Hello.
Hello. On which criteria?
Sound.
On which criteria does the
Hello?
FDA
Yes,
Hello?
sir.
Am I audible, sir?
Yes,
Yes,
audible.
right now. Right now.
On which criteria does the FDA classify NDA?
New drug application. Yes, because
sir.
suppose that this is not patented first thing. Second, it has proved its safety and efficacy. Both these things are required to establish that the drug is new by the FDA. If no patent is granted for the drug and it is a completely new to the market. These are the requirements of patent,
Okay,
particularly.
Okay, sir.
Patent is different, and patenting is different part. FDA comes into picture was the patent. This is particularly USPTO, which is granting the patent for a new drug, and FDA is giving the permission to market the drug.
Exactly, sir.
One more question from Priyanka ma'am. How to extend the expiration of patent?
Just I have told you that you can go for switching from particularly from prescription to OTC drug. Again, 3 years can be extended. Just you can go for, say for example, most of the drugs are marketed as the Resimates, but from Resimate you can go for a chiral moiety for either for R form or S form, and you can go for increasing the patent life of your drug.
Okay. Last question, sir. What is the hybrid of NDA, new drug
Hybrid,
application?
Hybrid new drug application, that is 505(b)(2). You can use it.
Okay, sir. Over to you, Zamale sir.
Thank you very much, sir, for this wonderful session. It was very informative one, where you have covered all the aspect related with new drug development, role of FDA, what is NDA, what is ANDA. I think what the knowledge you have shared today will definitely add, can say, new dimension to the knowledge of all the participants in respect to this regulatory perspective. So as we all are here on this virtual platform, and this is the best source to share the knowledge. So we take this particular platform to honor you, sir. So I request our principal sir to please felicitate the Dr. Alulkar sir with e-certificate as a token of appreciation. Sir.
Yes, sir. Yes, sir. Sir, thank you very much, sir. It was an informative session, and you covered almost all. I was scared that almost 68 slides was there, and I know you, sir. You have taught me. You are my philosopher, guide, teacher, everything. And I just know that you can defend on any topic. I know you, sir, from beginning, and it may be economics, it may be mythological, it may be scientific, non-technical, technical, anything you can talk on any topic. So I was scared, but you completed within time, sir. Thank you, sir. And it was such a wonderful session, and you can see the in chat box. It is full of questions, full of thanking this session. Thank you, sir. Thank you for this interactive and most informative session. On behalf of Srinath College of Pharmacy, IPA, Bharati Gramin Punarashna Sanstha, I congratulate and I appreciate you, sir. So this on behalf of BJPS, we present you, sir. Thank you, sir. Thank you for sparing the time.
Thank you. Thank you.
Thank you very much, sir.
Thank you.
I
Thank
hope
you,
this
all.
association will continue in future as well.
Okay. Thank you.
Thank you, sir. Thank you, Lord.
Now we will begin with the next session of the day. So this session will be coordinated by Ms. Poonam Sabre, madam. So I request madam to please take charge of this particular session and start the session.
Okay, thank you, sir. Am I audible to all of you?
Yes, ma'am.
Yes, ma'am.
Okay. Good afternoon and welcome you all for today's second session of National Level 1 Week E-Faculty Development Program on insight of basic techniques and recent advances in pharmaceutical education and research is jointly organized by Srinath College of Pharmacy, Aurangabad, in association with Indian Pharmaceutical Association, Aurangabad branch. Today we are having with us a very eminent resource person, Dr. Uday Devkate sir, and this task of introducing him is really a great privilege for me. Sir is presently working as an associate professor at Government College of Pharmacy, Aurangabad. He is having more than 24 years of experience in teaching and research. Apart from this, sir has more than 43 publications on his name and 1 patent to his credit. Sir has also presented at more than 40 various national and international conferences. Moreover, Sir has guided more than 20 MPharm students and 1 PhD student till now. In his 24 years of journey, Sir has held many important responsibilities. I would like to mention some. He has worked as a resource person in more than 20 conferences and faculty development programs. And Sir has also worked as an expert consultant at various universities like JNTU, SRTM Nanded University, Shivaji University, Nagpur University, and University of Pune. So with this wonderful and impressive introduction, I would like to request Dr. Devkate sir to please guide us on the topic entitled Basic Structure Elucidation, FTIR and NMR. Please, sir.
Thank you very much, madam.
Sir, before proceeding, I just want to make an appeal to all the participants. Please be with us because sir will be taking a little bit more time. Sir has requested, it was the session planned for 1 hour, but considering the task, sir has, I think so, it will take more time, and it is so interesting. Sir will be using the whiteboard too. So I request all the participants to be with us and be patient. Thank you.
Thank you, sir. Thank you, sir. I will not take that much time.
No, you take, sir. So we would love to.
Just I will complete within my given period. Thank you very much, sir. So I am very much thankful to Dr. Shoike sir and his team, Srinath Pharmacy College team, and also the IP Aurangabad brand for arranging such a novel FDP program and giving me the opportunity to deliver the talk. So my topic is somewhat different than whatever topic we have covered up till now. My topic is toward the structural elucidation. So up till now we have done certain theoretical part. So many of the people may not be interested to join or to listen this topic because they'll say that I'm not the chemistry fellow, so why should I come for the structural elucidation? But in all the thesis, okay, so we are using or we are having the NMR spectra, mass spectra, and we are using this IR spectra for the identification of the substances. So one part of my presentation is the interpretation of the IR spectra. So I am talking about the structural elucidation by using the various spectroscopic technique. So can you see my presentation here?
Yes, sir. We can see. Just make it slideshow, sir.
Yeah.
Yes, sir. Yes. Okay, sir.
Yeah. Thank you. So I'm talking about the structural elucidation. Structural elucidation using various spectroscopic technique. In brief, as a basis, I can explain you what is the role of various spectroscopic techniques in the structural elucidation, because not a single technique can be used to know the structure of a compound as a whole. We have to combine all techniques together to just know the structure. So we can start with the UV visible spectroscopy. UV visible spectroscopy, mostly we are using it for the quantitative estimations rather than for the structural elucidation. But some part of the structural elucidation can be done by using UV visible spectroscopy. It will tell you regarding the conjugation systems in your molecule. Then comes the IR that we are talking today, that is for the structure detection of the functional group which are present in your molecule. Then the molecular formula and molecular structure information can be obtained from the mass spectroscopy. NMR spectroscopy, if time permits, we can have some discussion on the NMR also. So NMR spectroscopy is used to, it is of two types, C13 NMR spectroscopy and PMR spectroscopy. So PMR spectroscopy can be used to detect the number of protons, environment of the protons. All about the protons can be obtained by using this PMR spectroscopy. And the C13 NMR spectroscopy tells you regarding the number of carbon and the environment of the carbon. So these are the all spectroscopic technique. All together can give you the information of the structure. But besides this spectroscopy, we also need X-ray spectroscopy for the 3D structural elucidation process. Orientations of molecule can be studied by using this X-ray spectroscopy. Then advancement of all the spectroscopies are there, like depth. Then we can have the 2D NMR spectroscopies that will give you the more detailed information about the molecule. Out of that, we'll just have discussion today, first with the interaction of this interpretation of the infrared radiations. Okay, so all this spectroscopic working principle is the interaction of this radiations with the molecule and what happened in the molecule. So that depends upon energy content of these radiations. Many of this radiation, depending upon their wavelength, they has the difference in energy, and depending upon their energy content, they interact with the molecule. All of we know that UV visible radiations, they are coming to us this side and having the highest energy content. X-ray, after X-ray means we can start from cosmic rays, gamma rays, then comes the vacuum UV, then comes the UV visible. So they are having the more energies, and as they are having the more energies, they can produce the electronic transactions in molecule, sigma to sigma strike, pi to pi strike. Such type of the transactions are possible, so they require the higher energies. After that, it comes the IR spectroscopy. Energy content is reduced, so they will react differently with the matter, so they will not produce any electronic transaction. But what they can produce, that they can produce the vibrational transaction. There is vibrational changes in the molecule, change in the bond length, change in the bond angle. Okay, so this is all theoretical part. We are not discussing this part here. Then comes the mass spectroscopy, where no interaction with the radiation is there. Still, we are using mass spectroscopy in the process of elucidation because the instrumentation is similar to the spectrophotometer. There is no interaction of electromagnetic radiation. The ions are separated according to their mass to charge ratio. Again, when we go to the next part, that is radio frequency radiations, the energy content is very less. It cannot produce any sort of the vibrational, rotational changes in the molecule.
Sir, sorry to interrupt, but
Yes,
your screen is not moving, sir.
not moving.
Screen is not moving. It is
No,
on
no,
the first slide only.
oh, I am just on the plus side only.
Okay, fine, sir. Sorry.
Okay.
It was question in the chat box that it is not moving, so that's what I thought it was.
No, I am on the first slide only.
Okay, sir.
Okay, so NMR spectroscopy, the energies we are talking, so it is very less, cannot produce any electronic, vibrational or rotational changes. So let us talk about the interpretation of the IR spectra now. This presentation is focused on the spectral analysis and interpreting the IR spectra rather than discussion on the theory of IR spectroscopy. Theory of IR spectroscopy, I will not discuss about the bending vibration, stretching vibrations. I will not discuss on the theory, Hooke's law, then simple harmonic motion, how to calculate, or what is the degree of freedom. Okay, so I will not discuss all this part because these all are theoretical. So all of you have studied this part in your previous classes. So we are going to talk about the identification of unknown compound by taking some examples. So we are just talking about the infrared spectroscopy. Infrared spectroscopy is the study of interaction of infrared light with the matter. Okay, so the fundamental measurements obtained in infrared spectroscopy is infrared spectrum. So we are doing the all determinations using this infrared spectrum. So we should know what is infrared spectrum, which is a plot of measured infrared intensity versus wavelength of the light. Means if you see the infrared spectrum on x-axis, you will observe the wavelength, and on y-axis, you will observe the percentage transmission. In UV, you may observe the absorbance on the y-axis. So we are getting a peak in the case of UV spectra, whereas in case of the IR spectra, we are getting certain inverted peaks. Okay, so every functional group will produce a inverted peak that due to the percentage transmission spectra. Okay, when a molecule absorbs infrared radiations, its chemical bonds vibrate. The bond can stretch, contact, and bend. This is why the infrared spectroscopy is called as a vibrational spectroscopy. Many misconceptions and facts are there about the IR spectroscopy. So IR is not generally used to determine whole structure of unknown molecules. It will give you only the very limited information about the molecules, that it will talk about the functional group. And just by knowing the functional group, not a single person can detect the structure. We don't need to analyze every single peak. It's used for identifying certain specific functional group like alcohol and carbonyls. Okay, so what is our method? So we have, our teachers has given the charts, and every time we are looking in the chart and finding the exact peak. We try to identify each and every peak within the spectrum. Okay, so that is called as a hunt and pick, is not the way to go. Trying to match every single pick to one of the numbers in the table. Okay, so a simple technique we will discuss here for the structural elucidation purpose. Before that, where we are using this IR spectroscopy. So that is what is the importance of IR. Okay, the sample or the spectrum is totally unknown. Many substances we are having, unknown substances are there, or even we are having the unknown spectrums. We can use this technique to identify the required examples include forensic sample, environmental waste samples, or new discovered samples where new material has been synthesized. Okay, in research we are using this part. So we are just taking the spectrum and we are comparing the peaks which produced in the spectrum. The second method or the second point where the sample or the spectrum is unknown and it needs to characterize or classify. It includes the example commercial applications where new additives or compounds are included, where the various additives can be identified. Okay, the sample mostly in many cases the sample is generally known, but the existence of specific chemical class needs to be determined. Example includes contaminant analysis, purity. For purity purpose, we are knowing the paracetamol is there, but what are the other impure substances which are present in paracetamol that can be detected? So all official methods has given this IR as a tool for identifying the purity of your bulk drugs. Okay, so now in our pharmaceutics or in many formulation process, we are doing some development of the formulations. We are imbibing the drugs with certain polymers, or we are doing some matrix imbibement, or we are doing certain changes in the molecule for the solubilization purpose. Okay, not whole molecule is changed. Some small modifications we are doing, keeping the molecule intact, and we need to confirm that the molecule is intact. Its structure has not been changed, and information can be obtained by using this IR spectroscopy. So sample is a complex known, and interpretation is required to confirm the material composition or quality, including product quality control also, and confirmation of structure of functionality of newly synthesized compound. What spectra or what part we can see in the spectrum? Okay, so either we are observing the peak intensities. Okay, so all of you know that peak intensities in IR, we are discussing about 3 types of peak intensities. One is strong peak, some are moderate peak, and some are weak peak. Okay, so this not only depend upon the structure, it also depend upon the concentration. So how much sample, because scarcity is of your sample available for the structural elucidation. Okay, even if your substance is having the intense group, but if you are giving very small sample for analysis, then it will be very difficult to identify the structure. So sufficient quantity of sample should be utilized. So this peak intensity will give you this information. Then origin of peak width, okay, generally depend upon the structure of the molecule. In general, the width of infrared band for solid and liquid sample is determined by number of chemical environment, which is related to the strength of intermolecular interaction. Okay, so we can handle liquid as well as solid samples. Many times we are developing the films in the laboratory, okay, transdermal dosage forms, and how much drug is imbibed in that particular patch of the transdermal system that we want to determine. Okay, so this IR can also be used for that purpose. It can be used for the quantitative estimations also. So this origin of peak width will give you the information how much area is there. Now, for the structural elucidation purpose, origin of group frequencies we are studying. Okay, so what groups are there within that region we are determining. Many functional groups absorb infrared radiation at about the same wave number, regardless of the structure of the rest of the molecule. Okay, so very little effect will be there on the neighboring group, but still ranges are given, not the exact values are given. For example, shear stretching vibrations usually appear between 2800 to 3200 centimeter inwards, and carbonyl C double bond O which appears between 1600 to 1800 centimeter inverse. This type of infrared bands are called as a group frequencies, and we are using this group frequency for the identification purposes. Before we go for the group frequencies, we should know what are the various regions of the IR spectrum. So by heart, we are doing this from our undergraduate, that there are two regions, fingerprint regions and group frequency region. The fingerprint region lies between 600 to 1450 centimeter inverse. Our teachers are saying the fingerprint region is a very important region. Every substance has a unique identifying characteristic, therefore it is called as a fingerprint region. But actually in practice, this is a very complicated part of your spectrum, and we cannot use only the fingerprint region for the structural elucidation purpose. We are not lying on the fingerprint region, but rather than we are using the group frequency region for identification of the structure, which lies between 1450 to 4000. So just I have developed a small single slide, okay, which will give you the informations of each and every peak which is producing in this group frequency region. So our basic aim is to study this part. Okay, so this spectrum you observe in your front, just it is drawn spectrum. So it is giving all peaks. Not a single substance is there which can show you all these peaks. Okay, so just this is the theoretical thing to remember. Okay, so it is not necessary to remember each and every part of your table. Okay, we can identify all important functional group in a single visual spectrum. Only a part of spectra we are considering is between 1500. You just see this 1500, 3000 centimeter inverse. Okay, so this part only give all information of the structure. Okay, so we are not considering this fingerprint region. Okay, many references are there for fingerprint region. Carburation of peaks can be studied by using this fingerprint region, but mostly we are using this part. Okay, the part which is from 1500 to 3000, and very small part after this 3000, which will help also for the structural elucidation. So we are just plotting two lines. We draw the lines at both the ends, and we are not considering the complex fingerprint regions below 1500. We will only oh, sorry, we will only analyze functional group region. Okay, so now we can see this every peak. Okay, step by step we can see. The first peak which we can observe is C double bond C. Okay, so it is between 1600 to 1680. Okay, it is a sharp needle-like peak we are getting. After this peak, a somewhat broader peak we are getting, that is of the C double bond O, which is lying between 1650 to 1780. Okay, so this is a finger-like, some broader peak is observed. Okay, so this gives the information about the C double bond O present in your molecule. Okay, this is followed by a tiny peak. Okay, this you can see these tiny peaks. Okay, like noise of the aromatic ore tubes. So this look like the ticks. Okay, so they look like the ticks. So they are generally present in aromatic. So these are very difficult to identify these aromatic overtones, because many times we may get confused with your noise. Okay, so for that purpose we are having the baseline correction process. Okay, so baseline correction only you will get more knowledge when you practice it. कधी baseline correction करायचं हे depend करतं की which substance is there. Okay, so some basic knowledge you need and some practice or some experience you need to overcome this baseline interference. Okay, so this, if you observe such ticks in your spectra, it might be due to the aromatic substances. Okay, so this is for the identification. The next peak, which is in the center of this part, is for the identification of two functional groups. Okay, if you are having C triple bond C, so you may get a peak between 2100 to 2260. Okay, so this peak will be for the C triple bond C type of the functional group. Similarly, at this 2220 to 2260 centimeter inverse, we may have the peak for C triple bond N. Okay, so that is for the nitrile groups. Okay, so this is also for identifying characteristic. After that, for every organic compounds, okay, you will get the one peak, okay, that is sp3 ch stretch, okay, for every, because every compound has carbon and hydrogen linkages, and for that we'll get this dog ear like structure, okay, ham, and that appearing between 2580 2960. Okay, so that is called as the sp3ch stretching. For every compound we will get such sp3ch stretching compounds. Okay, so immediately to the left of this, after 3000, okay, 3000 नंतरचा पण पार्ट थोडासा कन्सिडर करायचा आहे. तुम्ही म्हणताल नाही तर मी म्हटलं तुम्हाला की 1500 ते 3000, मग आता 3000च्या पुढे कशाला चालले, तर काही पार्ट तिथं सुद्धा आहे महत्वाचं. Okay, so this a sharp peak which we can observe here, that is sp2. Okay, so this is sp2ch stretch. So this sp2ch stretch, we have to confirm this sp2ch stretch by observing one peak at C double bond C. Okay, if you have one peak at C double bond C in the 1600 to 1680, the carburation of the peak is at this level. Okay, so presence of these two peak indicate or confirm the C double bond C alkenes in your compound. Okay, now after this, a sharp needle like peak. So these are not sharp peak. Okay, so these are the weak peak. I have not discussed about what you mean by weak and what you mean by sharp. So this is the intensity of the peak, how much length of the peak we are observing. So this is the short peak. It is in intermediate, it is medium. Now this is the SPCH sharp peak, SPCH stretch. Okay, so this is only available for triple bond containing. Okay, if your compound or your molecule contains C triple bond C attached with the hydrogen, so you'll get a sharp peak at 2700 to 3300 centimeter inverse. So this is again the carburation. So if you are having a peak at these levels and one peak at this level, we can just compare and we can identify or we can confirm that C triple bond C is present. Okay, so this is the simple things. After that, a very common peak which we are observing, that is OH. Okay, which is between 3200 to 3650. A broad peak will get. Okay, so it will a broad peak for the OH. So we can consider presence of OH, or we can just observe the presence of OH in this region. Okay, if you are having acids or amides, then also we will have to be considered. Okay, now see the next slide. A small additions has been shown by the different color. Okay, to just identify. Okay, so you may see, so if you are having the NH stretch, NH bend, okay, so this NH bend of amines and amides, that can be observed here in the range of C double bond O. So this, instead of C double bond O sharp group, we will have a broad bend of this amide group. Okay, NH bend. Okay, so this is the broad group which can be observed in this region. After that, to come to this part, okay, you can see a sharp peak. Okay, so this is the CH peak of aldehydes. So it is very difficult to identify aldehyde peaks as such, just by knowing, because our noise can interfere in determination. Okay, so this aldehyde functional group can be confirmed not using single IR, but it can be also confirmed. We have to combine this IR with NMR data, okay, to come for the particular. So this is CH bend, it is not stretch. Okay, so these are the bending vibrations which is there at 2600 to 2900 centimeter. A sharp peak you will get in this region. And beside this CH bend, we must have a C double bond O at 1700. Okay, if these two are there, it indicates that aldehydes are present in your molecule. Okay, so small things you can remember. From this we can come to a structural components. Okay, so after this, if the amines are present, NH, okay, NH bending vibrations, you may get the singlet or doublet or triplet. Okay, so this is splitted part, and it is broader, and in the region of the 3000, after the region of this more than 3000. Okay, so this is presence of amino group in your molecule that can be indicated by doublet triplets in this region. Okay, so a peak for aldehyde CH bed is also observed near 2600 to 2900 centimeter, which is supported by C double bond O stretch at 1700 centimeter. Okay, that can be confirmed by NMR spectroscopy. Now, I would memorize them how all of them are looking. Okay, just this single spectrum will give you information of all basic infrared groups which is available, and apply for the structural elucidation purposes. Okay, so we'll just have certain examples of the structural elucidation. Just see this. Now, I can't be able to hide this. Just I want to show the spectrum and then how we come to the conclusion that I had to show. Now see this one spectrum is in front of us. Now what we have to do, just draw the line from 1500 to 3000. Okay, and draw the baseline. Now the instruments are available which can draw the baselines. We can go for the baseline correction automatically. We have to set the limit and they will show the baseline corrections. Okay, so now you just see this spectrum. We don't have the C double bond C peak here. Okay, near the 1600. We don't have C double bond O peak here at 1700. We don't have C triple bond C or C triple bond N at 2200. Okay, so what we have, only one sp3. sp3 will be there common in all compounds because CH will be there and sp3 will be there in all compounds. After that, we are getting a OH broad peak. It indicates that it is alcohol. Just blindly we can say that our compound is alcohol. We will just discuss this with certain examples also later on. Now, one more example, or one more compound we can say alkanes. Okay, alkenes, ENE, double bond. So if you see the structure or if you see the spectrum, okay, so again I have drawn the line from 1500 to 3000. Okay, by using mouse, it was very difficult to draw this line. So I have already drawn it slowly. Online I can do it properly. So I had done it already before I prepare this presentation. Now see the baseline I had taken here. Okay, many times you may think that why not to take the baseline here. So this might be the C double bond C, C triple bond C. Okay, in the center 2100 or 2200. Then why I had taken this baseline here? Because only I want to talk about the alkenes. I don't want to talk about the other compounds. So see here at the 1600, I have this peak of C double bond C. sp3 will be common as CH will be present, and one more carburation of the peak will be there, that is sp2. Okay, after 3000 we are having a sharp peak here of this sp2, and this the presence of this peak indicate the presence of alkene in your structure. Okay, so this is the simple way how the alkenes are identified. Now see this complex spectra. Okay, again I have drawn a line from 1500 and one line at 3000. Okay, then I am just checking all the peaks which are present in this region. The first peak I can observe is C double bond C at 1600. Okay, so this C double bond C might be there. Then small aromatic overtones I have observed here. These aromatic overtones giving the information about the presence of aromatic group. Okay, so which can be confirmed by checking the peaks at this position 600. Okay, so fingerprint region also we have to check for these aromatic groups. As my discussion was not involving determination with the fingerprint region, I will only talk about the functional group region. Okay, so I am just observing this overtones. Again, sp3 will be common as CS stretch. After that, sp2 I will observe because C double bond C is there. So a small peak will be there of this sp2. There is no presence of hydroxyl group. There is no presence of the C double bond O group. There is no presence of C triple bond C. Okay, there is no presence of the aldehyde bending vibrations, no amides. So my structure is alkyl groups. Again, alkene, YNA, triple bond. Okay, so again a spectrum was in front of me. Now I had again drawn the line from 1500 to 3000, and I'm just observing. Okay, so this baseline might be from this part. We can draw the baseline here and see at the exactly in the center 2200. Okay, so we are getting, see, this is 2100 and 200 near 2200. We are having one peak of C triple bond C. This sp3 will be common. After that, a sharp peak I will get for the sp hybridization, CH stretch. Okay, this sp hybridization I will get. This is confirming the structure of C triple bond C alkene groups. So in this way, just by observing and drawing the lines, we can confirm the structure of the compound. Okay, so this is about the alkene group which is present in your spectrum. Now, after this, we will go for some examples. Just wait. Okay, now see the next spectra. So whenever we see the spectra, directly we just check the peaks. What is the problem? Simple problem is there. Which molecule is responsible for the falling spectra? And as per our discussion, directly we'll see the spectra. We'll draw the line in 1500 and 3000 and start detecting the structure of molecule. No, that is not the way. Don't jump to directly the spectral region. You just first check the molecule. Why? Because molecules are in front of you. Okay, omit the possibilities of the molecules by observing the functional group. Observe the molecule first. Don't observe the spectra. Okay, first job is to observe the molecule. You're identifying the characteristic of the molecule. Characteristic of molecule that indicated by the presence of functional group. Okay, so now see this first structure. There is a presence of C double bond C. Okay, so we should have a peak at 1600. Okay, see this 1600. Okay, so this is 1500. So this is 1600. So we don't have the peak at the 1600. Ultimately, this is not the possibility. See the other structures also. Here also there is CH double bond CH. Again, there is no peak at the 1600. Both the possibilities are vanished. Okay, so we can go next to the next part. Okay, here. So in this, what functional group we can observe? There is a presence of OH. Okay, we can see this presence of OH. And we also have a broad peak at 3500 for OH. Means the possibility of your structure is this. Okay, so you may have this functional group present in this structure may be there. Now you check the remaining structure. Here also we have OH. Okay, so this we can consider as we have this OH. Now in this structure there is ethereal linkage. Now from the structure it is confirmed that our substance contain OH. So we cannot go for OH here. Now we cannot omit the possibility of OH. And here in this structure there is no OH. So just we will omit this structure also. 1, 2, 3 structures has been omitted. Now these two structures are there, which is having the OH. Okay, now again see this both structure. Besides OH, this structure will also contain double bond O group. Okay, double bond O means that is near 1700. Okay, see here. You go for this 1700 region, there is no peak. Okay, again this possibility is also completed. So just this is the table. You just go through the table. Same thing what we had discussed in the spectrum, it is written on this table. Okay, so this all possibilities has been vanished and only this structure has been confirmed. Okay, we are able to confirm this structure by using the IR spectroscopy. Okay, this is very simple. So don't rush toward the spectra first. You try to see whatever possibilities or whatever structural components as given, whatever functional groups are present. We will take one more example for this structural elucidation. Okay, see this second structural elucidation. Which molecule is responsible for the falling IR spectra? Okay, so now you come one by one. We will discuss in the chat. Can we discuss it on the chat? Okay, so what you are observing in the spectra? Hello friends, please report your observation in chat, or you can unmute and you can talk. Yes, one is observing the C triple bond CH, but go for the first structure first. From structure 1 we can start. Yes, CH.
OH is not there in the group.
OH is not there. Yeah. So first structure contain OH as your one group. Then, so the possibility of the first structure as OH is not there. Okay, the possibility of first structure and possibility of third structure has been vanished. Okay, now this is presence of CH triple bond CH. Okay, so we may get the peak in the range of what? So this is sp3. Okay, this is sp3. It is correct. For every group, sp3 will be there. Okay, for CH triple bond CH, we should get a peak at 2200 here. Okay, 2200, 2100 to 2200. We don't have this peak here also. So this possibility is not. So this is also wrong. This is also wrong as OH is not there. Now you see this 2. Okay, so in both cases there is C double bond O and here also C double bond O. So you should get a peak at 1700. See this 1600, 1700. We are having a sharp peak, and this present shows the indication that C double bond O is present in your molecule. Okay, see this C double bond O here, C double bond O here. Now, which out of these two, how you will confirm the structure? Yes. दोघांपैकी कोणतं आहे आता तुम्ही कसं सांगणार? Anyone? You can unmute yourself or you can draw it. How to distinguish two structures? See here. Yes. This is very correct. See, this is to be drawn by someone that this is the CH bend. Okay, we are appearing this CH bend at this point. Okay, SP. This is SP3. After SP3, there is a presence of CH bend. Okay, that is of aldehydes. This is the characteristic of the aldehyde functional group. So we can just check that this is the possibility. Okay, because we are having the aldehyde bend peak at this level. Okay, so just by doing this, we can identify the exact structure. So this is not the possibility. See this. Okay, so this is the aldehyde which we are getting here, and all these possibilities has been vanished. In this way, we can have the simple structural elucidation process. Organizer may have. Hello. Shalke sir.
Yes,
Yes, sir.
अजून काही टाइम आहे का?
Yes, sir. आपल्याकडे टाइम आहे सर. आम्हाला नाही ऐकायचं सर आम्हाला.
नाही तेच म्हणत होतो मी. नाहीतर जर टाइम जर नसेल तर
नाही सर नाही. You take your own time, sir. We are with you.
But we will omit for the NMR because already it is 12:30. Okay, we will only go for the IR. Okay, so see here one more slide I have prepared. जे काय आता मी तुम्हाला एक सिंगल स्लाइड मध्ये सांगितलं होतं, तर स्टेप बाय स्टेप कन्फर्मेशन ऑफ स्ट्रक्चर we can discuss on this slide. See, this is a carbonyl group. Okay, carbonyl group present. First step is to see the carbonyl group. Whenever we observe we should say 1700. The groups between 1660 to 1820. If carbonyl groups are there, what are the possibilities for carbonyl groups? We have to check the possibilities of carbonyl group. Carbonyl group may be present in acids, it may be present in amide, it may be present in esters, anhydrides, aldehydes, or ketones. Okay, so this can be confirmed if acids are there. With your C double bond O group, we will have one peak for OH. There is presence of OH. Okay, so that presence of OH between 2400 to 3000 means after 3000 we should get a broad peak for the OH. Okay, so this is indicating the presence of acid. Then if amides are there, in case of amides, there will be a doublet or triplet present. Okay, it will be not sharp as the OH. Okay, so it is a somewhat broad peak will be there after 3000 centimeter inverse. If there is esters, okay, so esters can be confirmed by going towards the fingerprint region rather than the functional group region. Okay, for this OH, we have to see the bending vibrations in 1000 to 1300 centimeter inverse. Okay, if aldehydes are present, again, as we have seen aldehyde example, that there should be a bending vibrations of NH, CH stretch, CH bend at nearer to the 2700, 2900. Okay, aldehydes for this. Then in ketones, if we do not observe anything like this, okay, so it will be the ketone. Only you will get C double bond O in case of the ketone. If carbonyl groups are absent, okay, if C double bond O is absent, then we can go for the checking next possibility of the structural component, that either there may be the hydroxyl group. So this hydroxyl group can be checked by observing a broad peak between 3300 to 3400. Okay, confirm this with the CO bend in fingerprint region. We can also check it for the fingerprint region. So in amines, okay, so if in amines there will be the singlet, doublet. Okay, so you just see this single chart. Okay, so this single chart will give you all the information. If the amines are there, so you may get the peak here. Okay, with sites. If you don't have this C double bond. Okay, for ethers, ethereal linkage only can be checked into the fingerprint region. Okay, it cannot be changed into the functional group region. Okay, so then double bonds of the aromatic rings, if aromatic structures are there, we will get the overtones at the this level. Okay, so see these overtones, we can go for the aromatic, and with this overtones we should have this sp2 peaks. This presence of these two peaks will confirm the aromatic structure. Okay, then come the vinyl structure. Okay, so for vinyl we should get the more than C triple bond C. Okay, so see this, a sharp peak should be there after 3000. Okay, so this is for the presence of vinyl. Carburation of the peak, C triple bond C at this 2200 and 2100 to 2200, and one peak, a sharp peak at the after 3000. Okay, so this is the vinyl group. So then again we can confirm the triple bonds like C triple bond N and C triple bond C between this range. Okay, so again I given it in the here, C triple bond C and triple bond N. Just by understanding a single spectra, we means every peak will not be there in a single spectra, but this structure you can remember and you can identify any functional group in your molecule. So where you will require this. So now there is a time to write the thesis. Many students are working on writing the thesis and they are coming towards you, sir, this is my spectrum, let me know what are the functional group present. Okay, so what you can do, you have to just note down this important functional group in the table. You have to give the ranges what you observed, and you have to confirm this with the structure. Okay, so this will there should be the discussion also that my C double bond O C confirms with the original C double bond O C present in the molecule. It indicates that same substance I am using for the development of the formulations or my product is containing this, indicating the presence of the particular substances. Okay, so this presentation or this discussion should be written in your thesis, even if it is SUTIX for the confirmation of your, or for authentication of your drug, or if it is chemistry, if you're synthesizing new chemical entity or modifying it, then you have to write the structural elucidation in your thesis. Okay, so I think it is 12:30. Should I go for the NMR? Okay, sir.
Yes, sir. Yes, sir. Yes, sir.
Just I have to stop this now. How can I stop this?
So stop sharing, and the top of the screen will be there. It will be there. Stop sharing. Okay, sir. Now you can again share your
Second
next slide.
presentation. Yes, sir.
Yes, sir. Is it okay with everybody? You can message in chat box also. So accordingly, we can continue.
Okay.
Yes, sir. Participants are feasible. No issue, sir. We can continue, sir.
We can continue. I will just.
Yes, sir. Yes. Yes, yes, yes.
I will.
So again, you need to do share screening. Share screen.
Yeah, just wait. I'm opening this. कुठे गेला ते शेअरिंगचं?
खाली आहे सर. शेअर स्क्रीन हिरव्या कलरचं दिसेल बघा तुम्ही खाली बॉटमला. शेअर स्क्रीन म्हणून येईल खाली.
रिटर्न टू मीटिंग मध्ये जायचं का?
येस येस येस.
हा, शेअर स्क्रीन आला.
हा सर. स्लाइड शेअर करायचं. ओके सर. फाईन सर. Yes, yes, yes.
So just we'll have the discussion on the NMR spectroscopy. Okay, so up till now we have studied only the IR part. Now we'll discuss with the NMR spectroscopy, how NMR can be used for the structural elucidation purpose. Okay, before we go for NMR spectroscopy, small things I want to just discuss regarding the basis of the NMR spectroscopy. As the radiation does not contain too much energy that can produce the vibrational, rotational, or electronic changes in molecule. So what is there in NMR spectroscopy? There is only change into the magnetic properties. Okay, see the name, nuclear magnetic resonance. In all spectroscopy, we observe the resonance. So what do you mean by resonance? Resonance means something from ground state goes into the excited state. It will not remain long time in excited state. It will try to come back to the ground state by remitting back the energy, either in terms of the radiations or in terms of the heat. This is the fundamental principles of all spectroscopic technique. Okay, so resonance is involved. Resonance in case of UV electronic transactions are there. So there is also something from sigma to going for the sigma strike, that is excited state. Pi to pi strike, that is the excited state. In vibration also, there is something going from fundamental vibration to overtone vibrations, from one level of vibration to the other level of vibration. So what is there in the NMR spectroscope? Okay, so there is also absorption of these NMR radiations, and the nucleus, which is there, the spinning or the magnetic properties of nucleus, they are in the ground state, they will go to the excited state. Okay, and coming back to the ground state. Okay, so that is the basis of NMR spectroscopy. The resonance will be involved. Only the resonance is not there in the mass spectroscopy, because it is not the interaction of the electromagnetic radiation. In every spectroscopy, resonance will be there. Okay, so before we go for this mass spectroscopic problems, we should first know the ring rule. Okay, that is, if you have the molecular formula And from the molecular formula, how we can calculate the hydrogen ion deficiency, IHD. Some books says it as a HID, some books says it as a ISD. Okay, so one ISD is responsible for 2 hydrogen, means 1 double bond or 2 hydrogen. If there is a deficiency of 2 hydrogens, it will count for 1 HID. So we should know how to calculate this HID. I think all of you are knowing this, but as a part of this presentation, I will just summarize it fast. Okay, so there is a formula 2C plus 2 minus H, that is number of hydrogen, including halogen. Okay, 2C, that is number of carbon multiplied by 2. C2n plus 2, माझा एक simple formula होता, C2n plus 2 minus number of hydrogen including halogen. Okay, number of hydrogen including halogen, म्हणजे halogen सुद्धा जितके असतील तेवढे सुद्धा आपल्याला सबट्रॅक्ट करायचे, plus number of nitrogen. Okay, whole bracket plus number of nitrogen divided by 2. So it will give you the how many double bonds or rings are present in molecule, that can be calculated by using this formula. We'll just have some examples of this. We'll not go in how this formula is coming. What is the HID for each of the following molecules? See this, C6H10. Okay, so you just chart me what is the HID values. Okay, what is our formula? C2n minus number of hydrogen including halogen divided by 2. Okay, see this. First is your C6H10, number of carbon 6, 6 multiplied by 2, that is 12, minus hydrogen 10, that is 2 divided by 2. किती येईल? C6H10. Similarly, you go for C6H6, then you go for C4H8. Chart मध्ये टाका. Are you able to calculate fast? एक part of presentation आहे. See the calculated values I will write. C2n plus 2. साई दुने 12 plus 2, 14 minus 10, that is 4 divided by 2. Therefore 2. Okay, so in this way we can calculate the HID. So in NMR structural elucidation process, we first need to calculate the HID. Okay, so HID calculations are there. In some cases, if the molecular formula is not given, okay, if molecular formula is not given, only the molecular weight is given. In that case, we can determine this molecular formula by using rule of 13. Rule of 13 can be used to find out the possible molecular formula. So I will not discuss this rule of 13 here. Again, this is the example for calculating HID. We will omit this because it is taking time. Now see the first NMR problem. We have this formula C3H3Cl5, and we have these peaks. Only these two peaks we are observing. One is your triplet, one is your doublet, and they are appearing at the delta value of 4.52 and delta value of 6.02. Okay, and see this part. Okay, so this part is a ratio. In some spectra, this ratio is given in terms of the squares. Okay, where we can calculate. It's directly दिला me 1 hydrogen, 2 hydrogen. Okay, it might be given as a 3 squares and 6 squares. Okay, then we have to find out the ratio as 1:2, that is of the hydrogen. So now if this structure is, if this formula is given and it was asked to calculate the structure or to determine the structure, so what we can do? Okay, so we have to, first step is to calculate the HID. Okay, so what will be the HID of this structure? Please comment, anyone. हे कॉमेंट कशा पाहिजे हो सर? मी तुम्हाला तर म्हणतो कॉमेंट, पण मला दिसतच नाही कॉमेंट.
सर सर सगळे कॉमेंट करतायत चॅट बॉक्स मध्ये. खाली चॅट आहे ना, तर तिथे कॉमेंट करतायत सर सगळे.
ते कुठे बघायचं म्हणजे चॅट बॉक्स?
खाली चॅट म्हणून आहे सर, बॉटम लाईनला बघा सर तिथं. आता पहिले पण उत्तर दिले तुमच्या, तुम्ही जे विचारले ना 111 इथं आहेत बघा खाली. तिथं चॅटच्या
बाजूला आहे सर.
खाली शेअर स्क्रीन असते ना सर, त्याच्या बाजूला एक चॅट म्हणून येतं एक. मोर मध्ये वरती सर, वरती वर
मोर मध्ये बरोबर 102 चॅट येते.
हो हो.
बरोबर. HID. HID is 0. Yes, yes. Dalvi, Sampada Dalvi. She's saying HID is 0. Whatever. C3. Okay, 3 twos are 6 plus 2, 8 minus number of hydrogen, that is 3 plus number of halogen, that is 5. So that is 8 minus 8 divided by 2, 0. So first step is to just find out the HID. Okay, HID is 0. Then we have to write if the HID is 0, there is no double bond or no ring. Now you go for this ratio. Okay, so ratio indicate 1H. Okay, so it is a CH and 2H, that is CH2. Okay, so this ratio. Then you go for this splitting pattern. The first step is to find out the ratio. From the ratio, we can conclude that this is CH and this is CH2. Then the third stage is to go for the splitting pattern. You observe the splitting pattern. First signal is splitted into the triplet. So we have the formula N plus 1. N is the number of proton present onto the neighboring carbon plus 1. Okay, so that is the first step. It will give you the indication that how many protons are present onto the neighboring carbon. So triplicate indicates that my neighboring carbon will have 2 hydrogens. Okay, so this CH2. Okay, so this
फॉर्म आणलाय मी त्याचं काय फॉर्म हे फीसचं म्हणलं किती फीस होती त्याची
so the first triplet, okay, the first triplet says that my neighboring carbon
आणि ऍप्लिकेशन पाठव सांगितले
my neighboring carbon will have the 2 hydrogens. Okay, because this is the first triplet saying that my neighboring carbon has the 2 hydrogens. Okay, so this CH2 which is there we have determined here is confirmed. Then the second peak says the doublet. So what this doublet indicate that my neighboring carbon will have the 1 hydrogen plus N plus 1 rule is there. N is the number of hydrogen present onto the neighboring equivalent carbon plus 1. Okay, so this doublet indicate the presence of this CH. So this 2 CH and CH2 has been confirmed by this splitting pattern. So now we have what component that we have 1 CH, 1 CH2 out of this. Okay, so just we combine all the structural component together. Okay, so just we combine to get the various structures. Okay, so we have 3 carbons. So we'll just write 3 carbon here. HID is 0. So we'll write 1, 2, 3 carbons. Then we'll have 1 carbon as a CH2, another carbon as a CH. And we'll attach all the chlorines to this structure. Okay, so where to attach the chlorine? Till it satisfies the valency of carbon. Okay, so now see the structure and see the splitting pattern. Now this CH, okay, so this CH, the neighboring carbon contain 2 hydrogen will produce CH3, and this CH2, the hydrogen of this will produce doublet because neighboring carbon will contain the 1 hydrogen. Now we have to confirm this structure by allotting the delta values. See, these are the delta values. You can observe the delta values. Delta values are generally divided from 0 to 10. Okay, so this 0 to 10 values are characteristic. Nearer to the 0 is called as an upfield, and nearer to the 10 is called as a downfield. Okay, upfield appears for the shielded protons. The shielded protons means the protons which is not shared by the electronegative groups, which are not surrounded by the electronegative group, appears at the upfield, and the protons which are surrounded by the electronegative group will appear at the downfield. Okay, so now this chlorine is electronegative group. जर नुसता CH3 किंवा CH असतात, it should be appear at nearer to 1. Okay, if it is CH3, it should appear at 1. But now this CH, you see, it is appearing 6.02 because directly it is attached to the chlorine. Okay, so this doublet producing are attached directly to the chlorine. This it is attached to the carbon, which is again attached to the chlorine. So these electronegative groups, they produce the de-shielding effect. Okay, and this can be confirmed. We just assign this 1 and 2 values. So this is your triplet producing. This is 1 peak and this is 2 peak. In this way, we can elucidate the structure by using this NMR data. We will have the second examples for this again. You see, again, these examples are interesting. I think you may have solved this example at your undergraduate or graduate level, but still try to do this. The first step, as per our discussion, is determination of the HID. So what will be the HID for this structure? Chart. Again, same. HID will be 0. Yes, correct. HID is 0. Okay. Now the second step, check the ratio. Okay, CH3. This is CH3 and this is CH2. Okay, see the ratio and decide these two components. One is CH3, one is 2. Now you go for the splitting pattern. Okay, every splitting pattern says the singlet. Singlet means N plus 1, N is 0. Okay, neighboring carbon contains 0 hydrogen plus 1. So therefore it produces the singlet. Okay, so what this first singlet says, my neighboring carbon is without any hydrogen. So it is a simple C. This also says that my neighboring carbon is without hydrogen. So now we got 1, 2, and 3 carbons. Okay, we got 5 hydrogens. Now you assemble all these to get a particular structure. So when you are assembling, you have to arrange all the structures, okay, possible, and other structures also. Then we have to omit the possibilities. जे नाही आहेत ते बाजूला काढायचं, जे आलेत त्यांना tick करा, and you have to confirm the structure. Okay, now see, I have written this all carbon, carbon, carbon here. Then I have put this one CH3 here, then one CH2 here, and attach all chlorines. Okay, so see here, my both the peaks will produce singlet. This CH3 neighboring carbon without hydrogen, so it will produce singlet. This CH2 will also produce singlet. Okay, so I have 2 singlets here. The singlet can be confirmed by observing this structure. Okay, so now how to confirm the singlet? A singlet ह्याचाच हे कशावरून confirm करायचं? So this can be confirmed by alerting this or by checking this delta values. Okay, see the first delta value is 2.20 and the second delta value is at 4.02. Okay, 4.02 is somewhat more de-shielded as compared to 2.20. Okay, so see here, 4.20. This chlorine is directly attached to this CH2 group. Okay, so it is producing more de-shielding effect to this chlorine due to this chlorine. This CH3 is attached to the carbon which is attached to the electronegative group. याचा electronegative group याच्यापासून खूप दूर आहे. Okay, so they are appearing somewhat towards the upper side of your spectrum. So they are shielded protons. Okay, so these protons appears at the upfield and these protons appears at the downfield, and then we have to assign this as a 1 and 2. So I will just write 1 here and 2 here. In this way we can identify the structure. Okay, one more problem we can take. Now this is the interesting problem. Anyone, you can draw the structure here also. Okay, first you calculate the HID value. What will be the HID value? इथं डायरेक्ट लिहिलं तरी चालेल ना स्क्रीनवरती तुमच्याकडच्या पेननी बाजूच्या. What will be the HID value? C4H9Br. 4 2s are 8 plus 2, 10 minus 9 plus 1, 10. So HID value is 0. Yes. Then see the splitting pattern, the ratio. Okay, the second step is to see the ratio. The first ratio is 6 hydrogen. 6 hydrogen cannot sit on 1 carbon. Okay, so there might be the chances that these carbons, these hydrogens are equivalent hydrogen. So there may be two CH3 groups. Okay, so this will give you first information that I have two CH3 groups which is in the same environment. Okay, then this one hydrogen will says that I am CH. Okay, हे splitting pattern म्हणतात स्वतःबद्दल, हे रेशो म्हणतात स्वतःबद्दल, आणि स्प्लिटिंग पॅटर्न विल टेल यू अबाउट द नेबर. ओके, माझ्या शेजारी काय हे सांगणारे स्प्लिटिंग पॅटर्न, आणि मी काय हे सांगणारे तुमचा हा रेशो. Okay, so this ratio says that I am CH3 CH3, this ratio says that I am CH, and this ratio says that I am CH2. Okay, so this is the second step. Now third step, see the splitting pattern. Splitting pattern says doublet, means my neighbor contain one hydrogen. This says that my neighbor contain one hydrogen, so one hydrogen might be this. Okay, means my neighbor is CH, it is confirmed. Then this is multiplet. Multiplet is more than 5. If more than 5 hydrogens are there nearby, it is giving the multiplet. So if multiplet and singlets are there, it will be somewhat difficult to judge the neighbor. Okay, then comes the doublet. Doublet again, it says that my neighbor is CH. Okay, now we have 1 2 3 4, means all 4 hydrogens we got, all 9 hydrogens we got, all 4 carbons we got. 6 7 8 9 hydrogen मिळाले. Only we have to place the bromine. So you arrange these all 4 carbons. Okay, so we have 2 carbons with the same environment, so we'll just arrange CH3 here, which is attached to the carbon, which is attached to the CH2, and then Br. Okay, this is here only I can draw the known structure or the correct structure, but we also should go for the other nonsense structures also, and then omit their possibilities. Okay, by I can just write CH2 here, CH2, then again Br I can join here. Okay, I can write this CH3 afterward, and then I can say my peak is not multiplet. Okay, so that can be added. So see here how I am getting peak. This CH3, it attached to the carbon with one hydrogen. Okay, so this CH3 will give the doublet. As this CH3 is in the same environment, it will also give the doublet, and these two doublet will overlap on each other, and the height of peak will be more or equals to the 6th hydrogen. Okay, so this, therefore it is the ratio is of 6 hydrogen. Then comes the CH2. Okay, so this CH, the center one, it is having this CH attached near carbon, this CH attached to this carbon, and this CH2 attached to this carbon. Means there are 3 plus 3 6 plus 2 8 hydrogen plus 1. Okay, so that will produce the 9 8. Okay, nonate. So this nonate will be considered as a multiplet. Therefore this CH is giving the multiplet. Now this CH2 is again having one carbon which is attached to the hydrogen, and it is giving a doublet. So we are having 2 doublet, we are having 1 multiplet. So this structure confirms. Now you can confirm this structure by assigning these delta values. Okay, so this delta value can be assigned there. So now see the delta values. One is one doublet is at 1.02, whereas the other doublet is at 3.30. Okay, see this bromine, it is attached to this CH2. Okay, so this bromine is electron withdrawing group, causes it towards the downfield. Okay, so this third doublet might be this due to this CH2. Okay, and the first doublet might be due to this CH3. So we can assign this as 1 1, then this is the 2, and this is the 3. In this way we can elucidate the structure of compound using this NMR spectroscopy. One more structure we can go. Now see C10H14 having first singlet, second singlet at the delta value of 1.30 and delta value of 7.320. The first is equivalent to 9 hydrogen and the second is equivalent to 5 hydrogen. First you calculate the HID. Okay, you calculate the HID, how much it is? 8.
4.
How much HID is there?
It is 4.
हे 8 लिहिले कोणीतरी. It is 4. Okay, yes, it is 4. The HID is 4. How? 10 2s are 20 plus 2, 22 minus 14. Okay, 8 divided by 2, that is 4. The 4 HID, see, if you are getting the 4 HID, three things are there. If you are getting 4 HID, if we are getting one signal with singlet, which is lying between 7.2, okay, 7 to 7.5, if a signal is there, okay, and equals to 5 hydrogen. Okay, so this is all will make one identification that aromatic ring is there. Okay, directly it will solve the problem of all HID, because one aromatic ring, a pen कसं चालू होत नाही व्हायचं. बॉल पेन घेतलो. हे पेन चालू झाला असता ना, मला इथं स्ट्रक्चर काढता आलं असतं. पेनच चालू होत नाही. ओके, जाऊ दे. ठीक. Okay, so this 5 hydrogen, 7.2 delta value and singlet. This is all indication, and HID4. This is all the indication that aromatic group is present in your molecule. Okay, directly 10 पैकी 6 चा हिशोब मिटला. Okay, so just now remaining 4. Again, see here, 9 hydrogen cannot be sitting on one carbon. So all these are the 9 equivalent protons. So there may be CS3, CS3, CS3. Okay, so all CS3 will be here, 3 CS3s and 1 benzene ring. And as these are all giving the singlet, okay, the splitting pattern says that my neighboring carbon contain no hydrogen. Okay, so there is one carbon without hydrogen. Okay, so we have this structural component. So we'll try to combine all these structural component to get one structure. As we are having the benzene ring, HID4, we are having the benzene ring. So we'll just add the component to get the particular structure. Now see the peaks which are produced. So this CS3, 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 the neighboring carbon contain no hydrogen. So N+1 rule, it will split into the singlet. And definitely this aromatic ring, that is also giving the singlet. So this is only possible with the 1D NMR spectroscopy. I am not talking about the 2D NMR spectroscopy. In 2D NMR spectroscopy, benzene ring will give the 2 doublets. Okay, so that is different one. Okay, so just we'll have the basic version. So this is the 1D NMR spectroscopy where we'll consider it as a singlet. Okay, so this benzene ring is giving the singlet. So all 5 hydrogens, 1, 2, 3, 4, 5 are there on the benzene, and this all 9 hydrogens are there on this CS3. Okay, so now we have to assign la काय का दोनच peak आहेत. Okay, one is 1.30. Write this peak number 1, 1, 1 here, and write this peak 2 here. So the structure confirms. We'll have just one more examples. Yes, sir. Okay. First step, again, go for calculating HID. It was also C10H14. This is also C10H14. Yes, correct. The HID is 4. Now see the splitting pattern. The splitting pattern says it is 6 hydrogen. 6 hydrogen cannot sit on the single carbon. So all are in the same environment. So there may be CH3, CH3 here. Okay. This is the multiplet producing hydrogen. So this is CH. This is CH2. One benzene ring is there because our HID is 4. Presence of 4 HID. Okay. There is presence of a singlet and 7.12 peak with the 5 equivalent protons. This indicates that there is presence of aromatic ring. Okay. So this ratio we are consider. Now we'll go towards this splitting pattern. First peak is your doublet. Doublet indicates that my neighbor contain the 1 hydrogen. So this CH is confirmed here. My neighbor is CH. One multiplet where my neighbor contain the many hydrogen. Then this is again the doublet. My neighbor contain the 1 hydrogen. And this is the singlet that is of the aromatic. Okay. So we have all these components together. Now we assign all these component OH 9 त्याच्यात. OH नाही. Okay, so benzene ring. We will assign all this component to get a particular structure. इथं directly उत्तर देतोय मी तुम्हाला. It is CH2, CH, CH3, CH3. Okay, so we can have the other structures also. हा CH3 इथं सुद्धा आपण ऍड करू शकलो असतो. एक हायड्रोजन याच्यावरचा काढायचा, याला CH2 करायचा, याला CH आणि याच्यावरचा हे दोन्ही CH3 इथं शिफ्ट केल्यास. Okay, we can shift this CH3. Okay, so different structure you can think, but I am just short of the time. I am at the exact structure. Okay, now see the splitting pattern. This CH3 is in the same environment. Okay, so and the neighboring carbon contain one hydrogen, so they will produce the doublet. So these two doublets will overlap to each other, and the height of the peak will be equals to the 6 proton. So these are in the same environment. Now this CH, okay, neighboring carbon contain 3 plus 3 plus 2, 8 plus 1, 9. It will produce nonate. Okay, so nonate, that is a multiplet. So this hydrogen is also confirmed. Now this CH2, it is producing doublet. This CH2, the hydrogen, the carbon which is attached to this contain only one hydrogen. Plus n plus 1 rule indicates that there is presence of doublet here. Okay, so now you assign the values. Peak assign. Okay, कशाला कोणता peak म्हणायचा, कारण याच्यात दोन डबलेट आहेत. दोन मल्टीप्लेट आपण कन्सिडर करू शकतो. This is multiplet and this is aromatic. So how to assign these two doublets? One doublet is appearing at 0.88, whereas the other doublet is appearing at 2.45. Okay, how we can distinguish this? Sir, एक वाजायला लागला. Okay, see these two doublets. This CH3, it is attached to the carbon, which is attached to the carbon, which is attached to the aromatic ring. Means these CH3s are more shielded protons. Okay, these protons, CH3 protons are more shielded, so they will appear at the outfield, that is 0.88. Whereas this other doublets, okay, so the protons are appearing at the downfield due to this aromatic structure. Okay, due to this aromatic structure, they are going towards the downfield. So just we can write 1 here, then 2 here, 3 here, and 4 here. In this way, we can elucidate the structure. एक हजार interesting आहे. हा शेवटचा करू. Okay, नंतर just after this we can stop. Quintet, triplet, singlet. Okay, so just yes, HID is 5. तिथं दिसतं तुम्हाला आपोआपच आलं. पुढे yes, correct. HID is 5. The first splitting pattern shows, the first ratio shows it is CH2. The second ratio shows it is CH2. Anyone can draw the structure? HID is 5. 5 HID indicates that there may be one aromatic ring and one double bond. There may be one aromatic ring with another ring. There may be 5 double bonds. But as there is a singlet which is appearing at 7.17, it indicates that aromatic substances are there. Okay, now go to the splitting pattern. First step, HID calculation. Second step, finding the ratios. Third step, go for the splitting pattern. First splitting pattern is quintet. Quintet means 4. Okay, so 1, 4 will be there. If the neighboring carbon content, so it is quintet means 5. If the neighboring carbon content 4 hydrogen, it will produce quintet, 4 plus 1. Okay, the second is triplet. The neighboring carbon content 2 hydrogen, and this is the singlet, that is for the aromatic. Anyone can draw the structure. What is the possible structure here? Or just I will keep it as a suspense for you. Yes, benzene ring. Benzene ring मध्ये डबल बॉन्ड कशाला येतील? हे गोल केलं म्हणजे हा डबल बॉन्ड नाहीच आहे. Any more? One more chance. Anyone can draw the structure here. Or you just note down this. I will keep the answer in suspense. CH. पुढे. CH, CH3, CH3. CH3, CH3. मग पाचवी HID कुठे गेली? We are having 5 HIDs. ते स्ट्रक्चर नाही आहे. Okay, friends, I think I should stop here because already it is 1 o'clock. I am not giving you the exact answer of this structural installation. So you can comment or you can draw this structure or you can tell me the structure. Okay. One hint I want to give you, see the data given. Okay. The last peak, it is equivalent to 4 hydrogen. It is not equivalent to 5 hydrogen, indicating that it is a di-substituted benzene ring. It is a di-substituted benzene ring. Okay. So in the first case, it was a mono-substituted benzene ring equals to 5 hydrogen. In this case, it is a di-substituted benzene ring. So what will be the structure? We are having the 5 HIDs. So just you try to detect the structure. One important thing just I want to share with you. So whatever examples we are discussing here, okay, do you know where from I have taken all these examples? Anyone? Any guesses? Any book which you are knowing for the spectroscopy which you are using or for the determinations which you are using? हे कुठून असतील? कुठल्या पुस्तकातले? You will be surprised. You will be surprised that this is not from any Pavia, no. It is from Morrison Boyd, organic chemistry book. You just go for the Morrison Boyd and you check this. This is available in Morrison Boyd. Okay. So thank you very much. Thank you very much, the organizer, Sharke sir. You have given me the opportunity to present in front of all of you. Thank you very much.
Thank you, sir. Thank you. Karishma madam, any questions from your side?
Yes, sir. The first question is, what is the basic difference between FTIR and IR in respective of functional group detection?
Yes. It is very interesting.
By the next one.
Now see here, we are not having the simple IR instrument. Okay, we are all instrument today available are the FTIR, Fourier transform technique. Okay, so in Fourier transform technique, the intensity of the peak is increased. Okay, so this is done by using the interferometers. Okay, there is a interference produced in the path of radiations that will give the more sharper peaks. So today we are using the interferometers, that is FTIR instruments, rather than we are using the simple absorptive instruments. Old instruments where we are using the simple absorptive process.
The next question, where do we get peaks for NH-NH2 group in IR?
NH-NH2.
Dr. Ladda, sir.
Yeah, you just see this spectrum for NH-NH2. Okay, you will get a broad peak. Okay, which is covering near 1,700. Okay, so that is NH band, and one more peak is there that you will get near to about 3,000. Okay, we get one singlet, doublet, or triplet type of the peak we'll get. So that is for the NH band. So two peaks will be there. So amide groups, they can produce the peak in these two areas. You just see the slide which is given. Single slide will be sufficient to you. Okay, just you take the peak of that single slide, and that will help you to determine the structure of any functional group.
Okay.
Yes.
Next question. How to interrupt ortho-meta para positions of benzene ring by Hemlata madam.
Yeah, I can't hear you. What?
How to interpret ortho-meta para position of benzene ring?
Okay, so for this purpose, we had to go towards some finger point region. Okay, so in fingerprint regions at nearer 600 centimeter inverse. Okay, so you get a small peak. Okay, it will not be the sharp peak, so it will be a dented peak. Okay, so if it is a dented peak, so depending upon that shape, okay, so we can confirm. So benzene ring will get only the aromatic ham and the functional group region. So we just now I'm not going in more details of your fingerprint region.
Okay, sir. Next question is coming from Girvani sir. How to interpret cis and trans isomer?
Yeah, cis and trans isomers can also be distinguished. Again, we have to take the help of both fingerprint regions and your functional group region for detection of since these two. Okay, carburation of peak is the first answer for this.
Okay, sir. Last question. How to avoid noise levels while performing IR analysis?
Okay, background
Apart from
determination.
drying.
Yeah, it is very interesting. So many times people are not taking the background spectra. As we are not having the double beam instruments, we are having only the single beam instrument. We have to take the background spectra. Okay, either background spectra with air as a reference, or certain polystyrene films are there. We can keep this polystyrene film while taking the background spectra. Many times we are using DRS, and in case of DRS, we can take this background spectra by keeping the potassium chloride, dried potassium chloride in the sample cell, and then we can take the background spectra that will omit the interference. Okay, that will probably omit the interference of this clay light, and you will get the exact means. Again, some instruments are provided with the baseline correction facility, and they can automatically remove this, but determination of background is essential.
Okay, thank you, sir. Over to you, Poonam ma'am.
Okay. Now I would like to request our principal, Dr. S.D. Sheke sir, to felicitate our guest speaker, Dr. Devkate sir, with an e-certificate as a token of appreciation.
Yes, yes. So thank you, sir, for this wonderful session. As somebody has written that, I think so, Dr. Ladda sir has mentioned in the chat box that even a person from other specialization could also understand that. So that much in simplified way you have presented it, sir. And even I could also connect being a Sudix person. And I could claim that yes, I will be Atma Nirbhar now. But all the time when I am collecting the data and sharing the spectra with Damale sir, then I used to go with to Damale sir and ask him for interpretation. Then he used to interpret and used to calculate everything, and he used to give me the details regarding the compatibility study and all. Now I could claim and I could say that by listening this, I could say that yes, I could interpret and I could be more Atma Nirbhar now. Thank you, sir. Thank you. This elaborative session. It was explained in so informative and so simplified way. Thank you, sir. So, as an convener from the BGPS on behalf of Srinath College of Pharmacy, IPA, sir, we present you this certificate of appreciation. Thank you, sir. Thank you for sparing us time. Thank you, sir.
Thank you. Thank you for giving me the opportunity. First time I have given such type of the web seminars. Thank you very much.
Thank you, sir.
So may I give the conclusion remark?
Yes. Proceed, ma'am.
Thank you, Dr. Devkate sir, for this informative session. Today we have learned that how FTIR is used to quantitatively determine the concentration of molecules and how NMR spectroscopy is used to determine the molecule's chemical structure. Sir has explained very well about the important concepts, similarities, differences, and numericals related to these two powerful analytical techniques in such a simplified way that it will definitely going to be very helpful for all of us. Once again, thank you so much, sir, for this illuminating session. Now, on the behalf of Indian Pharmaceutical Association, Aurangabad branch, management, principal sir, and all the staff of Srinath College of Pharmacy, I would like to thank both the guest speakers of today's session, Dr. Alurkar sir and Dr. Devkate sir, for accepting our invitation, for sparing their valuable time and guiding us with their knowledge and experience. Thank you so much, both of you. And next, I would like to thank all the participants for attending today's session, and thank you for being such an interactive, attentive, and great audience. Thank you all once again. Now, without taking much time, I would like to request Mr. Manoj Damale sir to give further instructions for tomorrow's session. So,
Thank you, madam. Very warm good afternoon to one and all. And thank you for your active participation and patiently listening to both the session. So similarly, tomorrow also we have two sessions. The first session is of Dr. Ladda sir. He is going to explain how to do the extraction, isolation of phytochemicals. And followed that particular session, we have a second session of Gattani sir, where he going to discuss about how or what are the application of microbial flora in chronic conditions. The link of both the sessions will be shared in the evening on WhatsApp group. For today's feedback and attendance, the link of Google form is already been shared on the WhatsApp group. So I kindly request all the participants to please fill their attendance and feedback of today's sessions. So this is all about today's sessions. So here I conclude that all the session of today, day 2, has been over.
Okay, fine.
Thank you all.
Okay.